Раз. Раз.
Вообще шикарно же было. Вообще шикарно была презентация. Там люди смотрели, там вау, типа, особенно наши, которые. А вы что-то переживали там?
Ну, я в 6:30 с работы ушел 2 дня. Я в 6
Юра?
ушел, я
6 до утра работает 2 дня, да? Капец. Ну, блин, еще время дали? Да. Если время дали бы, то вообще, кажется, смысл было бы, да?
Я вообще как спринтерский забег. Я там предложение закончил.
Да. И, короче, я изначально сразу предложил, чтобы время увеличить, короче. Они говорят, нет, сейчас у нас времени нету. А в итоге там один спикер уже упал, и они успокоились из-за этого, и все нормально пошло.
10 минут. Что можно за 10 минут сказать?
Да,
только рот
интро.
открыть, да, и все.
И потом в конце сказать thank you for your attention. А так вообще хорошо.
Раз. Раз.
Да.
Okay, everyone, we have to continue our session. This session is about cancer biology, and I'm very happy to invite our old friend, Professor Gerry Melino, and he will be talking, as usual, about the p53 family of proteins, which are very important for tumor progression and many, many other things. Okay, the stage is yours.
Oh, maybe I use this one.
Okay, well, there's my microphone there.
This is to go forward.
Okay, the point is not working.
Okay. So I would like to go through some work which has been done in my laboratory for some time in order to leave a message. So I will talk about one protein in particular, p53, which is very well known because it's related to cancer. We'll see that in a moment. This is my disclosure. The main one is that I work for Springer Nature, and these are the journals that me, Ansu Vesimon, who is in the audience, and Mauro Piacentini, we have established in the last 30 years. So I'd like to share with you not only the usual view of p53, but some words. The first words that I would like to share with you is quality control. And this is because p53, as you know, is the most frequent genes that is mutated in any cancer. So this is a pan-cancer. In ovarian cancer, there is a certain degree of very, very high frequency of mutation, as you can see from the end. But in all cancer, about 50% of the human tumors, they need to physically inactivate p53, and another 20% to functionally inactivate p53 in order to allow cancer progression. So at least 2/3 of human cancer, they need to block these genes. And the mutations, so the structural inhibition is basically very specific in a certain point, certain area of p53. And this is shown, for example, also in some data. This is some work that we have published in PNAS some time ago. And basically, if we go in hypoxia with 1% origin, which is indicated by the arrow, we can see that in chromatin section, you have a very high concentration of p53. So although p53 is mutated, these are two of the most frequent mutations, it actually reaches the chromatin and transcription. And it does so by physically, in hypoxia, physically interacting with the IF1-alpha, and IF1-alpha is basically able to bring this mutated, in theory, inactivated tumor suppressor to the DNA. So basically, p53 mutated is able to hijack hypoxia-inducing factor 1-alpha and use that to actually have some transcriptional function. But that transcriptional function is not the normal one. And in fact, if we measure in some tumors the percent of survival, the ones that have IF and mutation p53, basically, they all fail to therapy. So this is how p53 actually is so involved in controlling of tumor suppression. So p53 is the gene that mainly controls the quality check of somatic cells. And why is that so? Because, we are exposed to environment. We are exposed to toxic environment. We spoke earlier on this morning about radiation, radon, and also toxic waste. And basically, there is a strong, important interaction between our genes that we have seen also today on the variation polymorphisms in relation to toxic environment. And our genes, they do interact with the environment. And there is a constant, basically, interaction between these two. And so toxic insults are really important for us to defend from. And these come either from endogenous, for example, free radicals, or from the exogenous, like a toxic environment. And to defend ourselves from this danger, we have established in the evolution a very specific set of defenses. It's not simply toxic means, I don't want any toxic effect. We do need some toxic effect, because otherwise we won't have evolution. We would not change. So we need adaptation and a little bit of adaptability to these responses is important for the human species. So this defense involves a set of genes which are depicted here. And P53 is really at the center of this game. And basically, P53 is one of the major, if not one of the most important genes in this response. It's not the only one, because in cancer, as I just said before, P53 is not functional. Still, we give toxic stimuli, radiotherapy, chemotherapy, and the patients, fortunately, they do respond. So there must be other mechanisms parallel to and independent of P53 to respond to this damage. And therefore, this stick with my first word that I'd like to share with you, quality control. P53 is one of the major quality control checkpoints for somatic cells. But P53 has two siblings, which we call for a general international agreement P63 and 73, although P53 is the molecular weight, but 63 and 73 is not the molecular weight, but we did like to do so. In fact, we demonstrated about 20 years ago, 25 years ago, that P73 is one of these ancillary pathways that is able to respond to DNA damage even independently, completely independent from P53. So not only P53, but also P73 controls to this quality control in somatic cells. This is some work that we published again some time ago, that hypoxia again is able to interact with P73. They do bind each other, and you can see that there is, in the presence of a P73, there is a hyper ubiquitination and degradation of IF1 alpha. In fact, in the absence of P73, hypoxia is able to change and modulate even more the vascular property of IF1 alpha. And indeed, in lung cancer, a specific isoform of P73 in the red line basically gives a protection against lung cancer as compared to the other isoforms. And indeed, in a single patient, we can see that there is an inverse relationship between the expression of P73 or the steady state level of IF1 alpha protein level. So it works in the, this isoform works in the opposite than the P53. Interestingly, the delta N P73, the blue line, works in the other direction. Here I bring another component in the equation, that P73 is not one protein, but two proteins, because there are two different proteins. And we did, in the last 20 years, analyze all the interactors, the post-translational modification, degradation, and transcriptional target of P73, which I don't want to go into the detail today. So the message is that not only P53, but P53 family members, all three of them, P63 I will show you later, they control the quality control of the somatic cells. So let's move together to the second part of my presentation about P63. And P63 is similar to P53, and we did some time ago a specific knockout on the one exon of P63, so the protein is expressed in the right time, at the right space, and in the correct amount. So it's like a knock-in in which we have replaced the C-terminus of these proteins. There are 3 different splicing variants at the C-terminus. So instead of expressing the alpha isoform, we basically switch to the beta isoform. This is a small technicality. A friend of mine, Frank McKeown, did publish data immunohistochemistry, not that one is mine. P63 is very similar to P53, but is on the ovaries. This isoform of P63 is on the ovaries, and therefore all the ladies, they have P63 from birth. And they have a P63 in a huge amount in the nucleus, in primary, secondary, and tertiary oocytes for 50 years. And this is what we found, not in the knockout, but in heterozygous mice. So the male fertility is normal. Female fertility is absolutely 0. There is no one single pups that came out from these female mice. And this is a strong evidence that the control of fertility in male and female is completely different. And one is P63 dependent, the other one is P63 independent. Why so? So to visualize with the confocal microscope that we saw before lunch, we crossed the mice with green mice expressing GFP c-KIT. c-KIT is stains pluripotent germinal cells, and therefore we can actually see this is the entire ovary, but we can see in green only the expression of these primordial germ cells. And normally, just a recap of embryology, at birth, the human female, they produce 7 million oocytes. At birth, within about 1 day or 2 days, half of them, 50%, 3.5 million, are physically eliminated. They are killed. And in a lifetime, you need basically these oocytes only to produce maybe 1 child, 2 child, in the old days 8, no more. But why you have to create 7 million in order to need only 4 or 5? Well, these days only 1. So there is a quality control on these cells that is incredible. And that the female, they have this quality control, then they are very messy, they are very disorganized, they have no quality control. So the quality control on the germinal cells that make sure that we have 4 limbs and not 6 is basically done through the female quality control and is P63 dependent. So in short, what happened is that the gonadotropic damage, chemical or radiation, basically makes some different type of damage, and this basically hit on the P63 that is sitting there, ready to kill, but it doesn't kill. It doesn't kill because P63 is a dimer, is a structural protein with 2 identical isoforms, and they basically they sit already on the DNA, as we saw from the histology. And you need a phosphorylation by the CHEK2, and then following that, there is a casein kinase that is ubiquitous that makes multiple phosphorylation. This phosphorylation, they basically block the conformation, they open the dimer, and the dimer, the monomer, cannot reassemble as a dimer, but they reassemble as a tetramer. The tetramer is the one that has transcriptional activity. The dimer has no transcriptional activity. So we see a very elegant change from a dimeric protein that is functionally inactive to a tetrameric that is active and kills immediately. And this requires the activation of the classic apoptotic BAX and PUMA. And basically you see that the protein from dimeric, this is a native gel, which is this lane, becomes tetrameric. And when it's tetrameric, it basically kills. And imagine that this is a lady undergoing chemotherapy for example, for a tumor. So this means that in order to preserve the integrity of the ovary and the oocytes, you have an area where you can intervene, not so much in order to give a future child, but basically to avoid basically the missing endocrine function of these organs. While we were submitted this paper, there was a German group who has identified in an infertility clinic 13 ladies with mutation with a premature ovarian insufficiency exactly on the exon 13, which is the sum, the one in red, that we have made basically the knockout. And so we tested the functional activity on PUMA and also on BAX. This, for the people who are doing transcription, if you have a five-fold is a strong induction. 1,000 is a huge induction. Eugene is looking at this. And so even this, that seems in a bit low, is about 150-fold induction transcriptional activity. So these are huge induction. So basically, what happened is that all the mutants, they have the tetrameric protein, the one that kills, and while the normal, the wild type, is a dimeric. So what happened is that this mutation switches automatically from dimeric and tetrameric and kills the oocyte during pregnancy. So that's why, not during pregnancy, in the ovary. So this lady, or the people undergoing chemotherapy, basically they have an empty follicles because they are eliminated by P63. So in summary, P63 is also, this was published a few years ago. Basically, P63 also is a quality control, but not on somatic cells. It's a quality control on germinal cells. So the word of quality control applies to the entire family. So we published with Arne Levine and Tak Mak many years ago that P53 controls the implantation of the fertilized egg and therefore contributes to fertility. P63, I've just showed you today, it controls really the formation of the oocytes. And also we published some time ago that P70 controls basically the quality of the blastocyst when it is formed. So this is a common trend of the entire family of controlling the somatic cells quality control and also the germinal. For the germinal, P63 is essential. It's interesting that in the equivalent of the genes family in the C. elegans, basically is called CEP1. CEP1 is very similar to P63 and is not present in all cells, CEP1, but guess what? On the meiotic post-spike 10 checkpoint. So there is a checkpoint of quality control of the oocytes, like the one I indicated at birth in humans, basically on the last step of maturation of the oocytes, there is the quality control of the germinal cells. Interestingly, CEP1, I just said, is like P63, and P63 is the ancient member of the family. I like to remember for some of the audience, especially the young one, this British scientist from Cambridge. He was JBS Haldane, and he identified that there is a cost of natural selection and that female, they have a very low mutation rate and a stronger quality control in the female, not male. So he knew that male and female, they have a different quality control, and he said there was a protective unknown factors. We would like to think the idea that P63 is such a factor. Read JBS Aldean because it's very interesting. Like during the Roman time with Titus, like in 1957, there was a crisis in the Middle East. The British, they blocked the Suez Canal, and he was so upset that he threw up his British passport, he left England and never went back, and became an Indian national, where he set up a very important genetic laboratory in what now we call Calcutta, on the east side of and is still very, very good genetic institute. He died as an Indian. So very interesting scientist. So what happened is that the word quality control applies to the entire family. And in P63, we have a quality control on the germinal cells. In evolution, we move to P53, that is basically a bone killer, and on somatic cells, on any cells. But also, and this is also the pathway that we identify on P63. So basically, P63 is a dimer, P53 is a tetramer. P63 is a very high concentration, inactive, and stays for 50 years on the ovocytes. P53 has a half-life of 15 minutes and kills straight away. And so high concentration, very inactive, close dimer, very low concentration. This is born to kill, P63, P53 is being evolved to actually make a very, very quick quality control. So to emphasize this, basically, P63 is the oldest member of the family, P53 is the young kid in the block. All of them, they control the somatic quality control, and P63, especially the germline. And there is a conformational evolution in this. As I said, from close dimer to phosphorylation dependent into a very short half-life tetramer. So there are structural constraints which has been evolved during this family of genes. Okay, so quality control, I'm insisting on this word, but are there other function? So we and Fred McKeown did a long time ago some work on P63 on the skin, and this is the keratinocytes, and it was a huge surprise that when you do the knockout of P63, there is no skin whatsoever. The skin in evolution comes much later because only mammalians, they have the epidermis like us. The other animal, they have cuticle, other isoform. And also, there is no limb. This mouse is like a sausage with a head, and you can see through the entire body. This is, there is absolutely even not a single keratinocyte. And it's not death, there is no cell death. So this is a huge mental shift, because up to now in the quality control, we just said, oh, is killing, is cell death, is programmed cell death. This is no death at all. This is a control of the staminal potential of a cell. So it's the same gene, you switch few things, you switch the promoter, and there are two promoters in the genes, and you, instead of killing, you do something else, completely unrelated. Is basically proliferation of the cells. And this brings me to the second word today that I would like you to remember, complexity. So you have one genes and you have a completely different function. So this is the latest work we've done on P63. We've done yet another knockout. It's the third one on P63, and we've done 4 on P73. And basically, we've done the elimination of the exon 13 like we did on P73. And basically, this mouse has it's a small. This is a real knockout. It's not heterozygous. It has a brittle hair and a very different skin. First of all, there is skin because the total P63 has no skin. So this one has a skin, but the skin is thicker. The skin is hyperproliferation. So the differentiation program of the skin is confused. The confusion is highlighted here. For the people working on the skin, keratin 5 and 14 are only on the basal layer. Loricin is only on the top layer, suprabasal layer. They never mix. And here you can see cells expressing a basal and suprabasal layer. So the differentiation program of the epidermis is completely confused and completely disorganized. And this is because P63, that isoform, basically lacks the ability to coordinate a more complex, and here's my second word, program. So we did some usual molecular stuff. We identified that there is a lack of cell adhesion, and this cell adhesion basically brings confusion in the program. For the people who want to read more, a few months ago we published in PNS a more detailed analysis of what I'm saying. So the genes as P73 is similar, promoter 1, promoter 2. One, basically, for P63 is only on the ovocytes, with one exception, and kills. The second promoter makes another protein, is on the epidermis, and controls the seminal potential. So by switching only few amino acids, the program, the interactor changes. And the C-terminus is important, as I indicated before, to coordinate the program. And so, what program? There was another friend of us, Paul Kawari, who identified a target of P63. It's called ZNF. It's one of the zinc finger proteins. ZNF, there are about 1,000 proteins, all transcription factor, and ZNF 750 was supposed to be a target. It is a target of P73, but to do what? So to do what, we've done our usual approach. We've done the knockout of this. And two things. First of all, the knockout has a skin. This mouse is different from the one of P63. There is a skin here. There is a skin program, very small, very defective, but there is a skin. Second, all mice, 100%, they die within few hours from birth. This is hours, 9 hours. So within 12 hours, they are all dead. And they die because they don't have permeability barrier. So the water retention is missing. This is from outside inside, and this is from inside out. So in any case, is completely defective. And so it controls the lipid biolayer, and it controls a set of genes indicated here, which basically control the entire lipid composition of the epidermis. And without lipids, there is no. So it's coordinated by P63, works in synergy with P63, but is delegated to do one specific thing. It controls about 12 genes. The way it controls these genes, this is a schematic from fatty acid, palmitoyl coenzyme A, keratosphinganin, down to ceramide, and very complex lipid in the formation of the skin. And basically, P63 controls the acid and neutral sphingomyelinase. You can see that the yellow, which is the knockout, is much lower. It controls also the DEG, the deacylglyceroltransferase, basically is completely absent, and the elevanoid. The elevanoid, they add from C20, 22, they go up to 28 and 36 carbons of lipids. They are all directly controlled by elevanoids, and there are 5 of them in the skin. And so basically, in a knockout MEF, if you reintroduce, or in a primary keratinocyte, if you reintroduce the P73, you restore the level and even at the lipid level, at the biochemical level. And this is the recognition of the binding site of all of these genes. So basically, the skin differentiation is delegated to a specific transcription factor that controls. So P73 controls ZNF750. We've done, as every good lab does, the bioID to identify physical interactors, and we found another interactors that is another ZNF, ZNF148. So we've done also the localization of this binding site in a human keratinocyte and also in head and neck cancer. P63 has 22,000 binding sites. ZNF148 has 40,000 sites, basically everything. And 5,000 are identical, they co-localize, and they co-localize on chromosome 21, which basically is the location site for D1, for the cell cycle control genes. And within the promoter of cyclin D1, there are 3 peaks. And these enhancer RNA are present in all head and neck cancer. And this is the mostly overexpressed and P63 mutated drivers in head and neck cancer. And basically what happened is that of the 3 enhancer number 3 is the one that recruits all the demethylases and produces actually an eRNA, which basically controls the overexpression of these genes, which is deregulated in head and neck cancer. Again, we published 2 months ago in July another PNS in which the story brings more details. So P63, I didn't talk about the E3 ligases, which is another story, quite complex. P63 controls the female fidelity, but is important for normal coordination of the differentiation program and has repercussion in cancer. So basically, the second word is complexity, because it's not necessarily the quality control and killing function, but the function could be quite complex. The same story I could give, but not today, on P73 and P53 doing a very complex interaction. And this maybe is the reason why it's not so simple to identify the function of P63. But what I'd like to say is that probably the maternal reproduction and fertility is the evolutionary ancient and most important function of the entire family, especially on P63. So the quality control of the germinal cells, which has been expanded by P53 to the quality control of the somatic cells. And then there's been more complexity to actually use that modules to do other things like differentiation and others which I'm not describing. And this function impinge on inflammation, immunity, and neurodegeneration. P73 is quite relevant for neurodifferentiation. So the third word is evolution. So P53 family modules is being adapted in different settings to do other functions which are evolutionarily distinct. And so creating an entire family evolution that is quite complex. So I would like to go through the story of P53 family today by highlighting the quality control of germinal somatic cells, the complexity of a multifunction of these genes, which is being conserved in evolution. So this is my last slides. Basically, the P63, P63, and P53, 63, 73, they are all important for the quality of the cells, and this module is being adapted for hippocampal degeneration, for neurodegeneration, and for epidermal stemness with a function unrelated to cell death. So we would like to thank my collaborators. First of all, is Tak Mak and Yufang Shi and Volker Deutsch, and also, he doesn't know, I consider a collaborator, JBS Haldane. So basically, we live in a difficult environment. We live in a different world. We have to learn to navigate and go through the difficulty in life. So thank you for your attention, and thank you.
Thank you, Gerry, for this very nice talk, but there's a lot to digest and think about. But for the sake of the time, I'm afraid there's no time for questions.
We have the discussion
yes,
during the coffee break.
Over the coffee break, I'm sure you'll have a lot of questions. So our next speaker is Professor Mauro Piacentini, another very dear friend of mine, and he will be talking about digital.
I think so. Thank you, Nick. So it's a great pleasure to be in Kazakhstan for the first time in my life. I hope it's not the last one. And I'm going to show you a story that actually we started together with Nick several years ago, and I hope now we have finished it because it's time to publish it. So most of the data are unpublished. Okay, so I'm going to talk about transglutaminase type 2. That is a very peculiar enzyme. As you can see in this cartoon, under physiological condition, it binds GTP is present in this closed conformation. When there is a stress for the cell in general, and the calcium is released from the intracellular stores, it binds several calcium ions, opens up, and exposes the active site, so became a transglutaminase. It's a complex enzyme because it can do many things according to its closed or open conformation and according to its localization in the cell. And generally, all the transglutaminases form cross-links, covalent cross-links among lysine and glutamine residues of proteins. But they can also deaminate the glutamine using the first part of the reaction. And this is very important in celiac disease because the gliadins that are the toxic component of the diet are made toxic due to the deamination operated by transglutaminase. But it can also incorporate amines into proteins, and keep in mind this because we are going to discuss this further during my presentation. In the closed conformation, it can act as a G protein, but also as a scaffold proteins. So, according to the many activity this enzyme can carry on, it was involved in practically all major disease, from celiac disease to inflammation, neurodegeneration, diabetes, and of course cancer. So I'm going to deal with cancer. And the first question, is transglutaminase relevant for cancer? These are TCGA data. And you see that when transglutaminase is low, the prognostic story is much more favorable. Okay, so high transglutaminase is a negative prognostic factor in many different cancers. So some years ago, we decided to start investigating the role of transglutaminase in liver cancer. At the time, there were no papers. Fortunately, there are still no papers on this subject. As you know, the hepatocellular carcinoma is the third most aggressive type of cancer. It kills more than 1 million people worldwide every year. And there are no real treatment for it, okay? So we decided to look in the literature, and we saw that also in this case, it seems that transglutaminase is a negative prognostic factor. In fact, you see the difference when you have high transglutaminase versus low transglutaminases. So how we approach the problem? We wanted to go in vivo, and so we use TG2 knockout mice. And we use a very old way to induce hepatocellular carcinoma, that is to give to young mice nitrosamine, that is a DNA damaging agent, and then you feed the animals for 34 weeks with the high-fat diet. This resembles a little bit more than other model what happens in humans. So although it's very long, we decided to apply this technology. And when we look at the mice, we were really, in a way, surprised, in a way, happy to see that the wild-type mice develop this huge hepatocellular carcinoma, but the knockout mice don't do so. And judging from the numbers here, you see that we never saw any tumor that was over 2 millimeters in knockout mice. They only develop, like the wild type, the preneoplastic little nodules that you see there. They are well known to hepatologists. In many cases, these little nodules regress autonomously. Of course, we look at the transglutaminase in the preneoplastic nodules, and you see that the enzyme is expressed in the preneoplastic nodules. That's fine, it's interesting, but you cannot knock out TG2 in humans. So we decided to use an inhibitor of transglutaminase, and the only available inhibitors is cysteamine. Cysteamine is a small molecule, you see it here, that is used for a genetic disorder that is cystianosis, that actually is a genetic disorder of the lysosomes. So it's used in children. It doesn't have major side effects, negative side effects. So we did the same approach I showed you before, but in the last 12 weeks, we gave cysteamine to the mice every day. And what we have seen is what you see here, that in this case, also the wild-type mice don't develop the tumors. And you see the numbers here. So inhibiting or ablating transglutaminase, you prevent the development of the hepatocellular carcinoma. You have only the formation of these little nodules that you see here. They are not real tumors. They are preneoplastic cells, actually. So we checked other parameters in these tumors. You see that TG2 not only controls the tumor development, also the inflammation. You see there is a quite remarkable decrease in many of the markers of inflammation, like IL-6, TNF-alpha, TGF-beta. And this is with the knockout, but also with the cysteamine, we get exactly the same result. And you see that the NF-κB pathway is downregulated in both cases. These are different tumors. You see that in both cases, the NF-κB is not really present anymore. So no tumors, no inflammation. So it seems quite relevant. If you downregulate transglutaminase in cell in culture, in this case in hepatocellular carcinoma HUH7, you see that the cell undergo typical apoptosis with the caspase cleavage, PARP cleavage. This is the tunnel staining. And what is interesting, I reported this data because not us, but another group using organoids showed that if you ablate TG2, you kill preferentially cancer stem cells into the organoids. Okay, so that are radically the cells that are present in the preneoplastic nodules I showed you before. Okay, so everything is interesting, but by which mechanism this is occurring? Okay, so what transglutaminase can do in the liver cells to prevent the formation of the hepatocellular carcinoma. And here there is an old story. So sometimes in science you miss an opportunity. Many years ago, actually in collaboration with Gerry Melino, we did one of the first interactome of TG2. We are talking about almost 30 years ago. And we found that transglutaminase can interact with nuclear histones. But we didn't believe to our data because at the time the enzyme was believed to be pure cytoplasmatic enzyme. So we neglected this data. But as sometimes happens, it's 5 years ago, this paper in Nature showed that transglutaminase can modify the histone H3. And you see, attaching covalently serotonin on this glutamine, okay? So this is a post-translational modification of the histones. And so this paper for us was quite shocking in a way, but I think opened up really an entire new area for the enzyme. And I show you why, because not only transglutaminase can attach serotonin on that glutamine, but can also remove serotonin and attach, for example, dopamine. And according to the amine that is incorporated, it can act as an eraser or as a writer. Okay, so really modulating the transcription and the activity of the histone H3K4. We verified that this is indeed the case. In fact, these are immunoprecipitation. You see that TG2 can interact with the H3KM 3 times methylated, but also with the one that is modified by itself, by the enzyme. And we showed that recently that through this epigenetic modulation, transglutaminase can regulate RIP kinase 3 and necropoptosis. So I told you that we believe for many years that transglutaminase was only present in the cytosol. Instead, this is not true. When you stress the cell, this is just one example with doxorubicin. You see that the enzyme leave the cytosol and move into the nucleus. Okay, so change completely its localization. And this is very important for its activity, of course. So in collaboration with Nick Barlev and Gerry Melino, we checked the status of histones in TG2 knockout MEF and TG2 and wild-type MEF. And you see that there are major differences here in the expression of the several histones in presence or absence of TG2. But not only that, also Nick did the analysis of methylase and demethylase, and you see that they are largely affected by the presence or absence of TG2. So a major activity that can modulate the chromatin. So if the enzyme goes in the nucleus and acts on the chromatin, you should expect that it has an effect on transcription. So what we did is to do the many things. First, we did the nuclear interactome of TG2 to see which are the partners in the nucleus of the enzyme. And then we also studied, I will show you later, the mRNA transcription in the presence or absence of TG2. Let's start from the interactome. So we identified and verified 91 proteins that specifically interact with TG2 in the nucleus. And we also have seen that this interactome is modulated according to the stimulus that you give to the cell. For example, if you give doxorubicin, 37 of these 91 are lost, and other 11 are acquired. Actually, there is also P53 here. Is here, so it's one of the interactors of TG2. But we got interested in these 4 proteins. Why? Because these 4 proteins are essential proteins of this complex, the CBAF complex. That is a very important complex that is important for chromatin remodeling. You see there are several components in this complex. Out of this, about 21, if I remember correctly, 7 are interacting with transglutaminase. And this complex is important to modulate the opening of the chromatin, so to make the chromatin, let's say, ready for transcription. And this is also interesting because we have shown, I showed you before, that transglutaminase can modify also the histone. So not only can modify the histone, but can also modulate the status of the chromatin. And these are some of the interactors of TG2. And just to give you an example, one of these, ARID1A, is important, the complex is important in liver, in the hepatocellular carcinoma. And you see that has a profile that is very similar to the one I showed you for transglutaminase. So high level of ARID1 is a negative factor for the hepatocellular carcinoma. And then we moved to the RNA-seq, and we did it both in the wild type and TG2 knockout mouse embryonic fibroblasts, and also in the tumors, of course. And here, the first, in a way, surprising data was that when you look at the maths proficient for transglutaminase or ablated for transglutaminase, you find 4,000 genes that are up regulated or down regulated. So a profound effect on transcription. And of course, we did an analysis of the pathway, and one of the pathways that was mostly affected is the Wnt pathway. As you know, the Wnt pathway is very important in tumors also. We also dissect, and you see it here. So these are the preneoplastic nodule, and then you see the Wnt beta-catenin pathway goes up when the hepatocellular carcinoma is formed, okay? And what is interesting is that when you look at the profile of the tumors, we saw that these in green are the wild-type tumor profile. And when you look at the transglutaminase preneoplastic nodule, you see that they are shifted over the normal liver tissue, okay? So their general profile is very different from the one that you find in the wild-type tumors. It's more similar to the normal tissue of the liver. And you see they are on the left side of this graph. And we found, I reported here about 15 genes that are upregulated in wild-type tumors, and they are downregulated in the preneoplastic nodules without transglutaminase. And we were very much interested in these two. Why? Because they are the key gene of the serine biosynthetic pathway. And you see that serine is one of the essential metabolites for the cell. So, of course, we were forced to study this pathway. Just to give you a recall of your biochemical studies, these are the two genes, PHGDH and PSAT1, and you see they have this, modified this component to form serine. But serine is also important because it also affects the GSH, so the glutathione pathway, and also the 1-carbon metabolism. So we look at the expression of these two genes in the tumors, and you see that these are 4 tumors in the ablated transglutaminase mice, this with Yasemin. You see that in both cases, these two genes are shut down. So there are no proteins related to them. So this pathway is completely silent upon inhibition or ablation of transglutaminase. So this prompted us to do the metabolomic, because if you have two genes that are important for the metabolism that are downregulated, you want to see what happens to the general picture of the metabolites. And so we did it, and what we found is rather interesting, that the ablation of TG2 per se in the liver tissue leads, for example, to the accumulation of these components that are the precursor of the serine. And this is even more marked in the tumors. And just the opposite occurs at the level of the glutathione. So the glutathione is reduced already in the liver tissue without transglutaminase, not tumoral, but liver tissue without transglutaminase, and is also more pronounced in the tumors. So what does it mean, this? That in the inhibition or ablation of transglutaminase, you don't have these two enzymes acting, and so you see the accumulation of the precursor, okay? And these affect the serine, but also the GSH and the 1-carbon metabolism, as you know, is essential for cell to grow because it's the basis for nucleotide metabolism and the DPH, GSH, and also for methylation reaction. So the absence of TG2 really changed the metabolism of this cell. We have other metabolites that are modified, but these are very important for this picture. And so the key question is, for example, HDG8 and PSAT important for the development of the hepatocellular carcinoma. as you can see, these are not our data, has been reported already that if you don't have a functional pH GDH, you have a hepatocellular carcinoma problem. And also, another interactor made by colleagues of us found that not only the G2 can regulate the transcription of this gene, but also interacts physically with pH GDH. And coming back to the histone story, it has been demonstrated by others that one of the genes that is regulated by the serotoninylation of the histone H3 is this one. So altogether, this picture seems to show that indeed this can be one of the ways by which transglutaminase can regulate the formation of the hepatocellular carcinoma. To sum up, this is a paper that was published some years ago, but it's an integrative genomic analysis of mouse and human hepatocellular carcinoma, and they define the pathways that are affected in the two conditions. We are happy to see that the Wnt pathway is one of the key pathways that is affected during the hepatocellular carcinoma development. The SYS SNF complex that I show you is one of the complexes that is modified by transglutaminase is also present. The methylase and the methylase, I didn't show PRB, but we published many years ago that PRB is a substrate of transglutaminase. So altogether, so many stimuli can induce the modification in the liver that are, in a way, preparing the liver cells to develop cancer. But the first things that is happening is the formation of these preneoplastic lesions. And to have that, it has been reported that you have to have ARID1 by chance, the Wnt pathway, P53, a PRB1 involvement. But then to go from preneoplastic nodule to real hepatocellular carcinoma, you need histone modification, chromatin remodeling, and finally, the upregulation of the Wnt beta-catenin pathway. And I show you that all these elements are in some way related to the transglutaminase. So what happens is that if you have TG2 in the tumor, you can develop regular hepatocellular carcinoma. If you ablate TG2, you have just the formation of these preneoplastic lesions that in many cases are able to regress autonomously. With that, I think I show you that TG2 modifying this histone, and I didn't mention one important thing. A paper appeared 2 weeks ago showed that cysteamine that we use in our work is able to block the serotoninylation of the glutamine 5. So the cysteamine is indeed an inhibitor of this post-translational modification. And the activity of the histone leads to the downregulation of these two enzymes, which are essential for the serine pathway and metabolism. And this is associated with the downregulation with the Wnt pathway and the inhibition of the hepatocellular carcinoma development. So we believe that the use of cysteamine in combination with the normal, let's say, treatment that are used for the liver hepatocellular carcinoma in clinics could be a good clinical trials to see whether we can improve the prognosis of these people. So this is my group. I already mentioned the collaboration with Nick. At the time, Nick and myself, we were in St. Petersburg. And with that, I thank you for your attention.
Thank you very much, Mauro. Thank you for being on time. But again, we have time for 1 or 2 questions maximum. Okay, while people are getting ready, let me ask you this question. So apparently, serotoninylation is very important of histone H3. But in addition to histone H3, there could be other proteins which undergo the same modifications. For example, you showed that some of the subunits of STILBAF complex interact with TG2. So my question is whether do you know if this modification is important, or this happens irrespective of TG2 enzymatic activity?
What we are doing now is to re-examine our interactons and looking for this serotonin presence or not in the interacting protein.
Yeah.
When we did it, we didn't know the story, so we didn't have done that. But we have the original data, so we can go back, and now with the mass spectrometry it's possible to see whether the serotonin is present or not. The other thing we are doing is to produce a specific antibody. There are specific antibodies for this post-selection modification to do immunoprecipitation of the serotonin-related proteins and by mass spec to see what they are and what they are doing.
Yes, and you can even do chipset after if you want to do
histones. Of course, you have to
histones.
verify if it's transcription or not, but we don't have to forget that transglutaminase is in the nucleus, but it's also in the cytosol. So it can also modify cytosolic protein attaching serotonin on the glutamine site of many proteins.
Of course.
And it is already known that there are many serotonin-related proteins in the cells. So one of these, for example, is the ROA that is very important for the cytoskeleton reorganization. So, lot to do.
Okay. Yes, well, Das?
Thank you very much for your very nice presentation. I just have two quick questions. Thank you, Dr. Piacentini. So, first question, what's the phenotype of knockout, TGA2 knockout? And second question is, when you're referring to the chromatin remodeling in carcinogenesis, in the tumorigenesis, what exactly you mean? So, because your markers you're talking about is mostly heterochromatin, methylation, this is methylation markers. Can you, it's activation, it's okay, opposite, okay. But what exactly you mean, like there are specific gene expression goes up, but what type of genes you think are behind this process?
Okay, so for the first question, actually I got the knockout mice from Gerry Melino. They don't have a specific phenotype, but whenever you stress these mice in any way possible, we did it in many ways, they start to develop a lot of problems, okay, of phenotypes.
But normal conditions,
in the cage, with the food and the water, they are okay, they reproduce normally, but if you stress them, you see many, many defects, okay?
Well, if you eat high-fat diet, because we all eat in Kazakhstan, then we'll be in trouble.
For the chromatin story, actually, we end up to do some epigenetic study, but we are not really expert of epigenetics, so we didn't have the tools to study whether the chromatin is modified in which way at the level of 3D in the absence of TG2, but other people, in addition to us, have shown that that serotonylation in placenta, for example, controls 8,000 genes, up-regulated or down-regulated. So seems to be a very important event that is also present during embryogenesis. So, of course, you have to do many chip assays. We did it for the RIP kinase 3, it works.
So last question probably. I think the Wnt pathway was maybe the first hint in your RNA-seq. Did you look into mechanism? Why is it specifically? Why specifically?
Okay, I didn't show you. I removed one slide because I wanted to be on time. Okay. So transglutaminase interacts with beta-catenin. It moves in the nucleus and lead to the degradation of F1. Okay? So we published that. So that's the reason I didn't mention.
Okay, thank you very much. And let's move on to our next speaker. I'm very happy to introduce you, Yasemin Sancak, who will be also talking about liver cancer, calcium metabolism in mitochondria.
Yes.
And we will be all excited. Thank you.
Thank you. I will continue the theme, but I want to begin by thanking the organizers for putting this exciting symposium together, meeting together, and for inviting me to celebrate with you. My name is Yasemin. I'm an associate professor at University of Washington in Seattle, and my lab's main focus is mitochondrial biology. And today, I'm going to talk about a recent paper we published that has to do with calcium signaling in pediatric liver cancer called fibrolamellar carcinoma. But before I dive into details, I want to tell you that metabolic adaptation is very important for survival. We are all sitting here, not engaging much, not moving around, and our metabolic demand is probably very low. But if, let's say, someone was to announce that there's coffee outside or dessert, and you want to run to the front of the line, you would start moving faster, which increases your metabolic demand, right? So it is very important, actually, to be able to switch back and forth between energy-saving mode and energy-producing mode. And this only doesn't happen in a whole body condition. This is also important for many diseases, for example, cancer. During tumorigenesis, cancer cells have to adapt their metabolism to accommodate the demands of growth that they face in the context of tumor microenvironment. So how does this happen? Well, at the heart of this metabolic switch is my favorite organelle, mitochondria. And you may know that mitochondria harbors 1/3 of all of your metabolic reactions, so it is very important for your metabolism. And inside mitochondria, I'm going to focus particularly on one protein called MCU, or mitochondrial calcium uniporter. This protein resides in the inner membrane of the mitochondria, and it facilitates entry of calcium ions from the cytosol into the mitochondrial matrix. And this MCU is very important for metabolic adaptation. How do we know that it is important? It is important because when we look at systems that do not have MCU, we see that they are unable to carry out processes that require a lot of energy. For example, MCU knockout cells do not grow as fast as their wild-type counterparts. Or MCU knockout mice are smaller. Again, they cannot meet the demands of growth. The knockout animals cannot run on a treadmill as well as the wild-type animals. And maybe not surprisingly, their mitochondria have defects. Their spare respiratory capacity is lower than wild-type mitochondria. So how does this happen at the molecular level? Why does MCU knockout have these phenotypes? So MCU is one protein, but it has many binding partners and regulators, and they all together form a protein complex, a channel that lets calcium inside the mitochondrial matrix. Once inside the mitochondrial matrix, calcium ions do a bunch of stuff, but the most well-known and most recognized is activation of the TCA cycle, which is the core metabolic pathway of the mitochondria. These three enzymes that I highlighted in blue are calcium-responsive enzymes of the TCA cycle, and they get activated by calcium. However, mitochondrial calcium is also associated with cell death. Here, in this image, you're looking at a mitochondrion on an electron microscopy grid, and this mitochondrion is exposed to high levels of calcium. As you can see over time, that calcium accumulates and it starts to precipitate as calcium phosphate deposits. So it forms these electron-dense particles. And after a while, that becomes detrimental to mitochondria. The mitochondria loses integrity. And if that happens in a cell, then the cell dies. Because high mitochondrial calcium is associated with cell death, we were actually very shocked to serendipitously find that in fibrolamellar carcinoma, mitochondria of fibrolamellar carcinoma is filled with calcium phosphate deposits. Here is again an EM image of normal liver, non-tumor liver, from the same patient. next to fibrolamellar carcinoma tumor, electron microscopy, you can see that if we quantify the number of mitochondria that has these calcium phosphate deposits, it's almost half of them. And this is in comparison to hepatocellular carcinoma, where we do not see this calcium phosphate accumulation. So this was very interesting, and before I go into detail, I want to talk a little bit about this cancer. FLC is a rare pediatric cancer of the liver, and it primarily affects young adolescents and young people, basically. It is very rare. In the US, there are approximately 100 cases detected every year. It has vague symptoms, it has known risk factors, and it has no biomarkers, and it has no effective therapies. So it is a very dramatic disease. There is one genetic driver of this cancer, and it is a deletion, genomic deletion, that fuses two proteins together, the DNA J heat shock protein with the protein kinase A catalytic subunit, making an oncogenic kinase, basically, that can be observed in the tumor samples compared to normal liver. So expression of this fusion kinase, which I will call DP, is the cancer-causing event in this rare cancer. Because we observed high calcium levels, we wanted to see if FLC shows high MCU expression. Again, it's a rare cancer. We do not have access to a lot of patient samples, but we are lucky to have access to some. And we looked at 4 patient and control tumor samples by Western blotting to see the levels of MCU and its binding proteins are increased. And you can see here that we see a significant increase in their expression. We can also see this in immunohistochemistry. Tumors have high MCU expression compared to non-tumor liver. And when we looked at publicly available gene expression data sets, we saw that MCU expression has also increased at the transcription level. It's not just protein level, its transcription is also increased. Unfortunately, there are no mouse models of FLC, and it's a rare cancer, so we can't get patient samples that easily. But we were, again, lucky to have access to cellular models of FLC, particularly using mouse hepatocyte cell line AML12. Someone has generated clones that using CRISPR that has the same genetic deletion to express the oncogene DNA JPKC fusion. And this cancer model is not ideal, but it's a good start, and it does recapitulate many of the phenotypes associated with this cancer. For example, these 2 clones that we have, C14 and C4, grow much faster than their wild-type counterparts. So we, of course, wanted to see whether we will also observe this calcium overload phenotype in this cellular model. To check that, we did this very simple experiment where we take mitochondria or cells and we treat them with an uncoupler, CCCP, which dissipates the mitochondrial membrane potential. And once membrane potential is gone, all the calcium stored in the mitochondria leaves the mitochondria. And then we can detect that mitochondrial release by using a calcium indicator dye. And when we do this experiment in wild-type AML12 cells and FLC models, we see that FLC models release a huge amount of calcium to the cytosol when they are treated with CCCP. And we, again, wanted to see, this is very interesting for us, and we wanted to see whether this calcium accumulation is a result of oncogene expression. To test this, we treated these cells with BLU compound, which is a PKA inhibitor. And to our surprise, we saw that the calcium overload phenotype that we observed in FLC cellular models was completely gone. Their calcium levels had reverted to almost wild-type levels. So it looks like FLC mitochondrial calcium increase is a characteristic of FLC, But we wanted to know whether there are any metabolic consequences of this increased calcium levels in FLC mitochondria. So in a parallel experiment, we were investigating how MCU, mitochondrial calcium, affects cellular metabolism. And we had done this RNA-seq experiment to see what metabolic pathways change in response to MCU loss. And on top of this list was this valine, leucine, and isoleucine degradation pathway. Valine, leucine, and isoleucine are known as branch-chain amino acids. They are essential amino acids. We have to get them from diet. And they have many very important functions in the cell. First of all, because they are amino acids, they are incorporated into proteins. Obviously, they are essential. But they also have signaling functions. For example, they can activate the mTOR signaling pathway. And their nitrogens can be used for generating glutamate, other amino acids, or nucleotides. So they can go into many biosynthetic pathways. And if they are degraded all the way down, those degradation products can enter the TCA cycle and again contribute to either energy production or generation of energy, ATP. Am I going forward? Okay. So BCAAs are basically very important. And when you knock down MCU in AML12 cells, we see that the proteins that are involved in this branch-chain amino acid catabolism pathway are all upregulated. So it looked like mitochondrial calcium signaling regulates this pathway. Excuse me. So this was very interesting to us because the BCAA pathway has been implicated in liver cancer and liver cancer tumorigenesis previously. So this was actually very, very interesting for us because the BCAA pathway has been implicated in liver cancer survival previously. And I'm not going to talk a lot about it, but suffice it to say that liver cancer suppresses this pathway. By suppressing BCAA degradation, they can save amino acids for protein synthesis and for signaling functions that will help tumor growth. And this pathway is actually a therapeutic target. If when activated, it prevents tumor growth. So I told you that loss of MCU leads to increased BCAA expression. So we wanted to see if FLC has high MCU levels, would it actually lead to decreased BCAA levels, right? So we looked at our patient data set, and we saw that the tumor cells, FLC tumors, had dramatically reduced expression of BCAA catabolism gene expression. So it looks like they are really downregulating this pathway. And we saw the same thing in publicly available RNA expression data sets. This also happens at the RNA level. FLC downregulates branch-chain amino acid catabolism pathway. And so it looks like MCU is upregulated and BCAA catabolism genes are downregulated in this cancer. And this is also recapitulated in the cellular models that we have. The BCAA catabolism goes down. And And of course, we wanted to see if this phenotype is dependent on mitochondrial calcium signaling. So we knocked down MCU in the cellular models of FLC, and we saw that the pathway expression goes up when MCU is knocked down. So of course, this is interesting. We can link this expression or changes in MCU, but does it have a phenotypic effect in this cancer? Again, we had to turn to our cellular models because there's no mouse models, and we wanted to see if we can activate BCAA catabolism pathway and see a phenotypic effect. To activate, we decided to overexpress the first enzyme in this degradation pathway, BCAT. And as you can see, we can achieve high expression of BCAT both in wild-type cells and in FLC cellular models. And when we did a growth assay, we saw that activation of this pathway had a dramatic impact in FLC growth, but wild-type cells were not affected, again, suggesting that FLC inhibits this pathway to save the BCAAs for potential signaling and for growth. Okay. So you may be asking, how come an ion channel regulates expression of this metabolic enzymes, right? We are talking about a calcium channel and then all the way to transcription regulation of this enzymatic pathway. So at this point, we took some clues from the literature. We knew that this transcription factor, KLF15, is very important for regulation of BCAA pathway genes in the liver. And when we looked at our patient samples, we saw that in the tumor, KLF15 levels were significantly reduced compared to wild-type cells. And we can see it in an immunohistochemistry as well. MCU expression and KLF15 expression are basically anti-correlative in this tumor when MCU is high and KLF15 levels are low. So we wanted to ask, does MCU actually play a role in KLF15 expression, or is it just correlation? So we showed that KLF15 levels are also low in cellular models, but when we knock down MCU in those models, we see that KLF15 expression goes up, suggesting that mitochondrial calcium regulates KLF15 expression as well. So we don't know the molecular link between MCU and KLF15, but we can, of course, start to speculate and follow up mechanistically. There is a lot of regulation of gene expression based on metabolic status. Many metabolites end up in the nucleus and then regulate the actions of different metabolic pathways and enzymes. And consistent with this hypothesis, we see that in FLC, levels of metabolites that regulate gene expression and epigenetics are significantly changed. So we think that MCU is actually contributing to gene expression changes and metabolic changes by regulating the levels of different metabolites, especially in this cancer, but maybe also in other contexts. So to summarize, we find that in this rare cancer, overexpression of DNA JPKC oncogene activates mitochondrial calcium signaling. And this cancer is characterized by increased mitochondrial calcium levels. This increased mitochondrial calcium levels is directly dependent on oncogene expression. And MCU regulates branched-chain amino acid catabolism. Urea cycle is something that we also found, but I don't have time to talk about today, through the transcription factor KLF15. And activation of BCAA catabolism inhibits FLC growth, and this could be a potential therapeutic target in FLC treatment. And with that, I would like to thank first all the patients for donating and their families for donating their tissues for research, all the funding resources and collaborators, and all the people in my lab who actually did this work. And thank you for your attention. I want to say I will take questions, but I don't know if I can talk anymore, so apologies.
Okay, thank you very much. I hope you will get better as a coffee break so we can take questions then. Okay, so we move on to our, should I do for you?
Yeah.
So the next speaker is Eugene Tulchinski from Nazarbayev University. And the title of his talk is Molecular Mechanism, EMT in bladder cancer, the impact of mRNA on alternative splicing. Should I give you a microphone?
This is not working. Okay, thank you, Nick. So firstly, I would like to thank the organizers for inviting and also for giving me opportunity to meet people I haven't seen for 6 or 7 years. So actually, my plan for today is just to show and to show the data which are in progress, or work in progress, and which are not published yet. I think it was much more interesting for me and probably for you as well. For this purpose, I even changed, as you can see, I even changed the title. I'm going to talk about ZEP1 and TAM receptors in bladder cancer. So my research interests are in the area of epithelial mesenchymal transition, mesenchymal epithelial transition, EMT and MET. So they are actually the basis of cancer cell plasticity. During EMT, carcinoma cells losing their epithelial appearance. There is a dissolution of intracellular epithelial junctions, such as tight junctions, desmosomes, adherent junctions, cells losing expression of ecadherin, which is epithelial marker. They acquire rear to front polarity, ability to migrate separately, dissociate from tumor mass, and activate expression of mesenchymal markers such as encadherin. Important point is that actually this process, EMT and MET, are not they're not like two defined states. So it's not binary process. Cells can pass through so-called hybrid states, which can be characterized as partial EMT or quasi-mesenchymal state. So these cells show quite high level of tumorigenicity, stemness, and they can actually play important role in metastasis and drug resistance. These stages in plastic processes occurring in carcinoma. What is important is that the key for plasticity, for cancer cell plasticity, are double negative feedback loops formed by transcription factors, so-called EMTTFs, and some microRNA species. For example, ZEP1 and 2 transcription factors, which can induce EMT, form double negative feedback with MIR200 family members. So this actually slide illustrates why it is applicable to study EMT in bladder cancer. You can see that all these Kaplan-Meier plots with EMTT. Why is it like that? Yeah, with EMTTFs show that high expression of of EMTTFs actually is a factor of poor prognosis for bladder cancer patients, but on the other hand, high expression of MIR200 family members is actually good for patient survival. So, meaning that when this equilibrium in bladder cancer moves towards more mesenchymal end of this line, then this is bad for the patient. So this is a figure from our very old paper published in 2009, analyzing expression of ZEP1 and correlation of ZEP1 expression, which is MTTF, with ecadherin in different carcinoma cell lines. You can see that there is actually nearly perfect inverse correlation in their level. People who work in the field know that actually ZEP1 is quite unique among other MTTFs. It is very difficult to find any system where ZEP1 would be expressed in epithelial tissues, suggesting that actually, meaning that ZEP1 is a bona fide inducer of EMT. ZEP1 gene is made of nine exons. ATG codon is located in exon 1, and the next ATG codon, which is in frame with this ATG, is located in exon 5. So the full size ZEP1, starting from exon 1 until the stop codon, which is in exon 9, is 1125 amino acids. The intron, the first intron, is huge. It is 150 kilobases long, and sometimes the extra exons, like exon 1 prime, for example, can appear in the messenger RNA, and this exon may contain, and there are several examples are known, are present in the databases, stop codon, which is in frame with the first ATG codon. So this messenger RNA would encode another isoform of ZEP1 with a length 906 amino acids. So it is shortened form. In some cases, exon 2 can be deleted due to, can be skipped due to alternative splicing, and also this would happen to frame shift mutation downstream of the first ATG codon, leading to formation of this second isoform. Another possibility, and this is also present in different databases, insertion of some some extra exons downstream of exon 1, which would generate isoforms with different N-termini. So the presence of different mutants, N-terminally truncated mutants of the ZEP1 are described and are available in databases. So ZEP1 is a zinc finger transcription factor with two widely distributed zinc finger clusters, N-terminal zinc finger cluster and C-terminal. There is a CTBP binding domain which interacts with CTBP core-rest and which is responsible for transcriptional silencing. At N-terminus, there is a NORT binding domain at very N-terminus and also PCAF-CBP binding domain, which is poorly mapped. So we don't know exactly when this CBP PCAF binds. Obviously, in truncated versions in the isoform 906, the feature of this isoform must be different because there is no binding of NORT and probably no binding of CBP P300 is possible. So we introduced, by CRISPR-Cas, we introduced knockout frame shift mutation in exon 2 which would actually mimic the situation where stop codon appears in exon 1 prime. When we did Western blots with different clones, so we did it in 3 different bladder cancer cell lines, T24, MUC3, EJ28. And most of these clones, but one, number 10 in T24, expressed truncated versions of the protein, or expressed some versions of ZEP1 which migrated faster than original isoform, which we know is present in wild-type T24 cells with a length 1125 amino acids. Then we actually introduced similar type of frame shift mutation in exon 6 downstream of the second ATG codon, and in this case, no ZEP1 was detectable in different clones in T24 cells, and also in clone 34, derivative of clone 20 of T24 cell line, expressing truncated version of ZEP1. Interestingly, only cells which express no ZEP1 whatsoever, in these cells, high level of EC-adherent epithelial marker and PIC-adherent, also epithelial specific protein, was detected. The activation of EC-adherent by knockout, complete knockout of ZEP1 was about 60-fold. However, in clones which express truncated forms of ZEP1, we could also find that these clones are quite different from original cell lines. For example, mesenchymal marker and cadherin was not expressed there, suggesting that these clones probably combine different features, epithelial and mesenchymal, and this allows us to hypothesize, and this will be checked in our future work, if this actually can be signs of hybrid EMT state. Another direction we are moving in is to how to interfere, how to target aggressive bladder cancer. And obviously, because ZEP1 is a transcription factor, which is very difficult to use drugs to inactivate it. So we looked at receptor tyrosine kinases, which are specific for mesenchymal cells and which are actually likely to be activated by ZEP1. And these are TAM family members, which are composed of 3 proteins, AXL, MER, and TYRA3. And actually, the normal function of this receptor tyrosine kinases is in efferocytosis. So they're expressed on the surface of professional phagocytes or non-professional phagocytes. And then, so these phagocytes, by using this receptor, they can recognize and engulf dying cells or apoptotic bodies, and actually then it will phagocytose them and consume. It is published that activation of AXL signaling could move again the equilibrium in cancer towards more mesenchymal state, and vice versa. Blocking AXL activity can move it closer to epithelial end of this equilibrium. So the idea of how actually AXL can operate in clinical settings is that if tumor is expressed to a drug, then this would obviously induce some apoptosis. Apoptosis would activate AXL, and again some hybrid or mesenchymal, more mesenchymal cells will appear in a tumor, and their survival will lead to formation of drug-tolerant persisters, which would generate, which is a source of tumor heterogeneity invasion, metastasis, and relapse. As we expected, AXL very nicely correlates with ZEB1 in TCGA project database, and also when we analyze expression of AXL in the cell lines, bladder cancer cell lines available to us, we found that it is expressed in mesenchymal cells and absent in epithelial cells. The expression of AXL was also detectable in cells showing hybrid features. These three cell lines co-express ecadherin, and vimentin, and also AXL. No correlation with MER or TYRA3 was detected. To test how targeting AXL can actually affect the development of, as monotherapy or combination therapy, can affect bladder tumors, we established tumor organoid technique in our lab in collaboration with National Oncology Center in Astana. And then we applied gemcitabine and cisplatin, so the common drugs used to treat bladder cancer, to these organoid cultures. And we found that at least in case of gemcitabine, there is activation of major isoforms of AXL and also TYRA3. The next step would be to check, obviously, how this influence features of these organoids or bladder cancer cell lines. For this purpose, we made single knockouts of TYRA and AXL. These are pooled population of knockout clones shown here. And then also double knockouts of both of these genes. Surprisingly, we found that in case of their knockout, there is increased level of MER, the third member of TAM family receptors. The triple knockout is already available to us, made by our research assistant Sandugash. So this is all ongoing. Interestingly, what I also wanted to show here, we did treatments we treated T24 cells with TAM inhibitor, ITP0903. And so we cultured these cells either in 2D cultures or as organoids in matrix gel and in organoid media. And then after treatment with this inhibitor and RNA-seq, we found that all 10 main hits in organoid cultures are cell cycle related. So this is all about cell cycle. Strangely enough, no EMT process was detected here. But when we culture cells in 2D system, there was a completely different response. This is all metabolic processes. And also EMT was detected here, which is possibly what people report when they use 2D cultures and treat these cultures with AXL targeting drugs. So our future perspectives is to characterize the one isoforms and try to find out whether there is a link between them and hybrid or quasi-mesenchymal states in carcinoma, in bladder carcinoma. And also we're looking at, we're doing now the analysis of applicability of TAM receptor tyrosine kinase inhibitor for bladder cancer treatment as monotherapy and in combination with gemcitabine and cisplatin. And obviously we have all the cell models with different knockouts of these two as controls. I would like to thank Nusum, my colleagues, the groups of Marina Krajewska, Nick Barlev, Dieter Rittmacher, with whom we collaborate. Also, I'd like to thank Dmitry Gushin, who worked here. Unfortunately, he left. He's not with us any longer. But he invented these CRISPR-Cas techniques at Newsom, and we're now broadly using this technique. And also, I would like to thank Sanjar Shelikenov, who is our clinical colleague from NROC, from National Research Oncology Center. Thank you for your attention.
Thank you very much, Eugene, for being on time. We have time for one question. Please. Okay, so I will have a quick question. Okay, you're saying that EMT genes, they confer drug resistance.
Yeah.
Okay, EMT state, I should say. Okay, so what would be the, yeah.
It is just common knowledge.
Yeah, so in case of those receptors, do you know the mechanism? How would they do it?
Yeah, I think there is a lot of biological sense behind this, because when macrophage, for example, or dendritic cells recognize dying cells, this happens usually in toxic environments. Cells are dying around, and so the activation of these RTKs results in activation of pro-survival mechanism via PI3K mostly.
Okay, but the simplest mechanism to tolerate cytotoxic drugs is to do the cell cycle arrest. But here it's a different situation. It's proliferative.
It looks like it is, yeah. No, there are a lot of reports on. I mean, the problem here with this inhibitor, as with all other type 1 RTK inhibitors, is that it's not specific.
Okay.
I don't know which other kinases it inhibits. Probably CDKs or whatever.
Yeah, so it's a very complex effect. Okay. All right, thank you very much. We have to move on because we're running out of time. And I have to announce myself in the next 15 minutes or so. I will, before the coffee break, I will tell you the story which is not finished yet, it's ongoing. And this concerns the mechanisms of CAR-T resistance that tumor cells develop during the treatment. So what is the CAR-T cells? CAR-T cells are T cells engineered to express chimeric antigen receptor that can bind target cells independently of the classical TCR MHC1 pathway. Therefore, they become versatile tools to kill any type of cancer cells. So what is the CAR, chimeric antigen receptor? It is shown here. So it consists of antigenic binding domain and intracellular costimulatory domain and T-cell activation domain. So upon activation, T-cells would secrete granzyme, perforin, and cytotoxic cytokines such as interferon gamma and TNF alpha that would induce so-called immunogenic cell death. The pioneer in this research was Professor Carl June from University of Pennsylvania, and in 2012 he participated in the treatment of a young girl, Emily Whitehead, who was successfully cured with CAR-T technology with this very aggressive type of leukemia, which is acute lymphoblastic leukemia. And that was a great success. But despite the great success of CAR-T technology for hematological cancers, the success for CAR-T in treating solid tumors is not very impressive at all. And there are many reasons to that. Number one, tumor immune microenvironment. It could be tumor heterogeneity, suppression of antigen presentation, shedding of the antigen, and low penetrance of T cells through extracellular matrix. So we know that tumors are able to suppress immune cells by various types. They also very heterogenic, so the expression of the antigen could be very high in few cells, and then it could be suppressed in other cells because of different reasons. And on top of it, this MHC, which presents this cancer epitope to T cell receptor, cancer cell can down-regulate the expression of MHC, so T cells will not be able to recognize that this cell has to be destroyed. In addition to this, there is another mechanism of antigen shedding. That's when tumor cells express proteases that cut off the antigen from the surface. So T cells have no idea whom to kill and why to kill. So again, these tumor cells can escape the immunogenic cell death. But however, I have to tell you that CAR T and activated T cells are able to kill tumor cells not always through direct interaction. They can kill, it's called bystander killing, by expressing those cytotoxic cytokines, TNF-alpha and interferon gamma, without direct contact through MHC and TCR. So that means that tumor cells should be able to develop an intrinsic mechanism how to survive this effect, bystander effect of T cells. And having this in mind, we set out to identify new genes that protect tumor cells from the effect of T cells. To do this, we used DCAS9 activating screening system. So when CRISPR-Cas, everybody knows what it is, and when I'm talking about CRISPR-Cas screening, everybody thinks, okay, that should be knockout. No, it's not knockout, it's opposite. Okay, so we're looking for genes that are not repressed, that are activated. So how we do it? We use mutant Cas9, which is not able to cut DNA, but it's perfectly able to bind DNA. So this DCAS9 is fused with very powerful transcriptional activators, such as VP16 and HSF and P65. So this makes this very powerful synthetic activation, transcriptional activator that can be targeted to any locus by specific gRNA, sgRNA. So we used the library which was developed in the laboratory of Ijang, and we took HeLa cells, which is a very popular system. We infected this library along with DCAS9 fusion protein, and we also made these HeLa cells to express CD19 antigen. I have to tell you that CD19 is expressed only on B cells. Okay, it is not expressed on HeLa cells. The reason for that, we do this because we wanted to avoid any potential antigen shedding, which I told you a few minutes ago. So we took those HeLa cells, infected with the library and DCAS9 activator, and incubated with CAR T cells that specifically recognize CD19 antigen on HeLa cells. And of course, most of the cells would die, but some of them will still survive. So we took those, we proliferate them, and incubated with CAR T cells again. and again. So we repeated 3 times to identify those cells which are resistant to CAR T therapy. What we also did, we did sequencing after the first round and after the last round. And then we compared the results, and they're very similar, which is good. So we also repeated the screening in vivo in mice. So we took immunocompromised mice, injected with HeLa cells that express CD19 and the library of DCAS9 gRNAs. And of course, those mice which were not treated with CD19 CAR T, they developed huge tumors. Now, those mice, 2 groups that were treated with CD19 CAR T, some of them responded and the tumor shrank, but the other group did not respond, so tumor grew up. And that was the most interesting group. So we sequenced those, and we were looking at the enrichment, the functional enrichment of gRNAs that were pulled out from those mice. And it turned out that there are 3 groups of connected genes, which are shown here. The first group is about mitochondria. The second group has something to do with cytoskeleton. And the third group is connected to oxidative stress. So that means that there are 3 potential mechanisms how tumor cells can cope with the CAR T toxic therapy. So, what we decided to focus our attention is the first group, mitochondrial. In particular, we were interested in this gene, which is called AK4. Very similar to the AK-74, you know what it is. It's a machine gun, right? So this is a very interesting name for the gene, and what it does, it uses the phosphate of ATP in the reaction. It's a kinase, it's a adenylate kinase. So what it does, it phosphorylates AMP and makes it ADP, adenosine diphosphate. So basically, from 2 molecules, it makes 2 molecules of ADP. And the main sensor in the cell of ATP amount is AMPK, AMP kinase. And there is a connection between AK4 activity and AMPK. Okay, if you look at the prognosis of AK4 overexpression in different tumors, you can see that if AK4 is overexpressed in pancreatic cancer, cervical cancer, renal cancer, stomach cancer, overexpression of AK4 is associated with poor prognosis of cancer patients. So that means that AK4 is a potential oncogene. Now, here the upper part, you see that wild-type HeLa cells, after the treatment with cytotoxic cytokines, they die eventually after 75 hours. This is the blue line. And the green line are cells without AK4, and they die much faster. Now, the red one are cells that overexpress AK4, and they seem to be very resilient to the effect of those cytokines, TNF and interferon gamma. Below is another experiment showing the apoptosis and XN5 staining. And here, in the absence of AK4, you see increased apoptosis. And the lowest amount of cell death we see in cells that overexpress AK4. So again, it shows that AK4 is important for survival of tumor cells in response to immunogenic cell stress. Okay, so what we did next, we did RNA sequencing to look for signatures. We were not looking for individual genes, which I think it's stupid, because the whole assay is not for individual genes, it's for signatures. So we were looking for signatures of genes that were altered in cells that overexpress AK4 versus cells that underexpress AK4. And what we found, the most interesting signatures, and most obvious ones, were hypoxia, which is not a big surprise, TNF-alpha pathway, oxidative phosphorylation, EMT, interferon response, because we treated those cells with TNF and interferon gamma, so TNF pathway and interferon gamma pathway should come up, and they did. In addition to that, we saw glycolysis pathway to be activated. Okay, so to really confirm these interesting signatures, we did profiling, real-time profiling using seahorse. Okay, I want to spare your attention from those very complicated data. You just focus on this two bars. I'm talking about spare respiratory capacity. What is a spare respiratory capacity? It's an indication of the fitness of the cell. So the higher SRC is, the better cell feels. If the SRC lower, then cells are about to die. So you can see that after the treatment with those cytokines, both control cells and knockout for AK4 cells, they don't feel good, they're about to die, but those cells that overexpress AK4, they survive, and the fitness is higher than in the other two cases, which is very interesting. Now, to get to the more physiological conditions, we decided to focus on colorectal cancer, and we analyzed a bunch of cell lines that we had as a collaboration with Hans-Uwe Simon, who will be talking after the coffee break. And we chose several lines, and what we did, we analyzed those cell lines for the amount of ATP and phospho-AMPK. Why ATP? Because I told you before that AK4 decreases the amount of ATP by phosphorylating AMP, right? So in theory, if you remove AK4, you should increase the amount of ATP. And that's exactly was the case in all cell lines that we tested. ATP level goes up in the absence of AK4. And in addition to that, phospho-AMPK, which is the indication of AMPK activation. So the phospho-AMPK level goes down. That means that AMPK is not active. And I have to tell you that AMPK is one of the major regulators of autophagy. Just to remind you what autophagy is, in under conditions of low energy, AMPK becomes activated, phosphorylated, and it modifies another kinase which is called OOC. And OOC with VPS34 and other kinase, they mobilize phosphoinositol 3 phosphate to the membrane, and then that's how invagination starts. And then LC3 becomes lipidated, and then VP P62 comes in and brings the cargo to autophagosome, and then autophagosome fuses with lysosome, and then autolysosome destroys all the proteins inside the vesicle. Okay. So the obvious question was, okay, if we have AK4 overexpressing cells, will we see increased autophagy? And the answer is yes. You can see here that in cells overexpressing AK4, you see highest signal of LC3, lipidated form of LC3, compared to control cells or knockdown AK4 cells. That means that AK4 promotes autophagy. And you may ask me, okay, how autophagy interferes with immunogenic cell death? And the answer to this is that on many, many different levels. For example, through CIAP, through RIP kinase, through activation of procaspase 8, which requires a lot of ATP. So if you don't have ATP, you don't have cell death. Sorry, I have to take a sip. Okay, good. So what we show so far is that AK4 promotes autophagy that may interfere with immunogenic cell death. Another experiment which was done in another lab came out very handy. So there is a small molecule inhibitor which is called autophagy. So autophagy inhibits VPS34 kinase and blocks autophagy. So they took HeLa cells and HCT116 cells, which is another cell line for colorectal. Both of them are insensitive to TNF or trail. But after incubation with this inhibitor, they become sensitive. So, for example, activation of caspase 8 goes from nothing to very robust levels. The same is true for HCT116. So this means that the inhibitor of autophagy would synthesize tumor cells to immunogenic cell death. And we also tested our favorite cells, RKO and LOVO, which are colorectal cancer cells. And in each case, you can see that the absence of AK4 makes cells more sensitive to killing mediated by TNF and interferon gamma. And if you overexpress in HeLa cells, if you overexpress AK4, they become more resistant to both interferon gamma and TNF alpha treatment. Okay, I want to finish my talk here. A lot of work has to be done before publication, but I think this is a very interesting study. Why is it interesting? Because first, we show that autophagy, a very important cellular process, interferes with immunogenic cell death. And second, AK4 may work as a predictor for success of CAR T therapy. If we take patients, we analyze for the expression of AK4, we can predict, okay, these patients will be more sensitive to CAR T treatment, and those will be less sensitive, and then they have to use inhibitors of autophagy to make good progress. And to finish my talk, I want to acknowledge Alexei Petukhov, who is a PhD student in my lab. That's mostly his work, and other people in the lab were helping him, Shingiz, Alina, Aliyah, and others. And also I want to thank our Mazov Research Center in St. Petersburg in Russia, and Hans-Uwe Zimmermann and Nikita from Institute of Pharmacology, Bern, in Switzerland, and other people from Institute of Cytology in Russia. Okay, with that, I will want to finish. And because we ran out of time, if you have any questions, I will be happy to answer during the coffee break. Thank you. If you guys want to stay here, I'll be happy to answer questions.
Dear colleagues, the coffee break is moved to upper atrium, and then we have half an hour for the coffee break, and we will be waiting everyone here by 17 p.m., 5 p.m. for next session. Thank you.
How do you feel?
Сейчас, подождите.
I don't know, because this resists CAR-T.
Yeah.
Right? Yeah.
No, but because there are a lot of, when solid tumors are treated, there is a fail of treatment.
Yes.
Do they describe that there is the autophagy is there?
Yes, they were, yeah.
Я один раз даже отказалась от ведения, от руководства, потому что моя смаялась с докторантом, пока она в декрет уходила. И такое мне показывало, я говорю, нет, дорогая, все. Или ты пишешь, как я скажу, или делаешь, как положено, или отказываешь. Ну, она ушла спокойно, была акушером гинеколога, у нас на стыке было. И ты спокойно, уже годы спокойно все.
Ну да, бывает, что поедете на ВМК, а если чуть-чуть подведено, можно еще...
Ну и акушеры, они более спокойно ко многим вещам относятся. Но я зато, ну просто иной раз понимаешь, что себя Там еще так все заряжает, вот это все что-то. Ты хочешь, не хочешь, вовлечешься. Пришел себе, да я сижу. А потом как начинаешь, как сколько они защищают. Хоккей такой довольно агрессивный.
Check.
Are you the chairperson? You are the chairperson. That's most important. Chair the session.
No.
I will see. I tried. I know we are late.
Yeah.
For the slides.
Yeah.
And on microphone.
Раз.
Yeah, it works.
Раз.
Lucas, maybe this place closer, okay? Because of its time, it seems to me 3 minutes we should start, not wait for after. Let's wait a few hours.
It's too empty.
Yes. Because of it's too far. The coffee break, please. From Марина Алексеевна, вы не хотели бы поближе? Да. Все собой.
уберу свои вещи.
А уже?
Уже все убрал, убрал.
Құрметті ханымдар мен мырзалар, конференция қатысушылар, жұмысымызды жалғастырайық. Dear ladies and gentlemen, dear conference participants, let's continue its last section. It's an honor to conclude the first day of the conference. Okay, let me introduce our keynote speaker and of our last section, Microbial Communities and Immune Responses, Professor Hans-Ove Simon, President of the London Medical School, University of Bern, Switzerland, with the topic Neutrophils and Extracellular DNA Traps.
Yeah, dear colleagues, thank you very much for the introduction. I also would like to congratulate you here in Astana to have built such a nice research institution. Congratulations not only on the success but also to your birthday and to bring us all here together in Astana. So I'm very happy to be here. Regarding the content of my talk, we switch a little bit the topic, but you have heard already in the last talk that also in cancer research, the immune system is very important. So I think in the last session of today, we are talking about the immune system, and my topic is neutrophils and extracellular DNA traps. So what are neutrophil extracellular traps? These are extracellular structures with a DNA scaffold, and on these DNA scaffolds associated are granule proteins, which are very toxic against pathogens. And when these extracellular structures were discovered, this was a new concept, and therefore it gained a lot of attention in the community. So far, it was believed that neutrophils kill bacteria intracellularly after phagocytosis. So the new concept was that neutrophils can also kill bacteria in the extracellular space. So you see here, sorry, one cannot see it. Here, these are neutrophils. These are neutrophils, and they release these DNA structures. These are bacteria, and these bacteria are captured, therefore they call it trap. So they trap the bacteria, and subsequently these bacteria are killed in these extracellular DNA-containing structures. So this discovery was done in 2004 by the group of Zielinski in Berlin, and we were a little bit later, and we published with different agonists a similar observation. So we are from the very beginning, so we work now for more than 20 years in this field. So, and soon after this discovery, people were thinking, so how can these DNA-containing structures enter into the extracellular space? So most of the people guessed that somehow there must be cell death in the neutrophils in order that the DNA is getting out. While we have observed that viable neutrophils, is on this side, can generate these DNA-containing structures, and when you then compare the results, what people described, how these structures are generated, you see major differences. For example, you see here elastase, and people use knockout, so one would say, okay, these are same conditions, so here they say elastase is important, we say elastase is not important, PAD4 is important, PAD4 is not important, RIP kinase 3 is important, RIP kinase 3 is not important. So the solution for these contrasting data is that maybe we describe different biological phenomena. So this we call neutrophil cytolysis, and this we call neutrophil extracellular trap formation. So there's a confusion in the literature because people think that after this neutrophil cytolysis, the neutrophil extracellular traps are also generated, but the morphology of these structures are completely different. So in this case, we see a DNA cloud, and in this case, we indeed see DNA fibers. And I want to show you also how these DNA fibers look like. So we basically investigate just this mechanism. And here you see neutrophils. So we stained here the nucleus, and in green you see these extracellular structures. So these extracellular structures are generated by viable cells within less than 2 minutes. You get this, and the cells remain viable. So these structures then can bind bacteria. It's shown here. The bacteria are labeled with green color, so we can see them, and then subsequently these bacteria are killed. So we do these functional assays. We always prove when we talk about neutrophil extracellular traps that these DNA-containing structures can bind and kill bacteria. So this is a summary, the knowledge of 2019, how these neutrophil extracellular traps are generated. This is a neutrophil. You see here many receptors. All these receptors, after activation, can lead to neutrophil extracellular trap formation, which is the result here. So, and the take-home message here is what you need is an activation of the NADPH oxidase, which generates reactive oxygen species, and you need an active cytoskeleton, so actin polymerization as well as tubulin polymerization. And why do you need that? Because you have to translocate granules and mitochondria to the leading edge of the neutrophils. And then somehow these neutrophil extracellular traps are formatted in the extracellular space. And in order to get everything, you need an additional stimulus of energy. And so you see here ATP, and the ATP you get by glycolysis in the cell, because neutrophils are very weak in oxidative phosphorylation. So we still indicated here the mitochondria, because you need the mitochondria also for glycolysis, because you need here the NAD plus for glycolysis. So the next question was, many of these receptors, they can trigger calcium. So the question was, what is the role of calcium signaling for the generation of these neutrophil extracellular traps? And we started to answer this question. We started a very simple experiment. So we cultured the neutrophils in different concentration of calcium, and you see, if you culture the cells without calcium, you are not able to generate such neutrophil extracellular DNA traps. And if you have too much calcium, you also cannot generate these neutrophil extracellular traps. So you need something here between 1 and 2 millimolar calcium, and this correlates to the physiological calcium concentration. So extracellular calcium seems to be important or required for the formation of these neutrophil extracellular traps. So for what do we need extracellular calcium? This was when I was a postdoc. You see, after stimulation of neutrophils with platelet activating factor, you get some calcium response, even in calcium-free medium. So this is mobilization of intracellular calcium. And when you have extracellular calcium, the signal is much larger, and it's also longer. So you have basically more calcium, sustained calcium signaling in the cell. And for such sustained extracellular calcium, you need potassium channels. So you need a potassium efflux in order to get such extracellular calcium in the cell. So to understand now which potassium channel is important, we used a genetic approach. So we used different knockout mice, isolated the neutrophils, and just checked whether the neutrophils can still generate these neutrophil extracellular DNA traps. And you see in most cases, in spite of knocking out certain potassium channels, neutrophils can still do it. But in this case, it's a BK channel. So if the neutrophils have no BK channel, they cannot generate neutrophil extracellular traps. So we got interested in this BK channel. So we analyzed the calcium response in these neutrophils, and you see that the neutrophils without BK, they have less maximum calcium levels, and they have also reduced sustained calcium influx, and in calcium-free medium you see no difference. So when you talk with people who work on ion channels, they measure currents. So we started also to measure currents, and you see in BK containing neutrophils as well as BK knockout neutrophils, you can measure currents in the cell. But if we use now known BK inhibitors, we get a reduction of the currents in those cells which contain a BK channel, but no difference in the BK knockout neutrophils. And this is a second BK inhibitor, and you have same results. So that indicates that neutrophils have indeed a functional BK channel on their surface. So the BK channel is important for neutrophil extracellular traps. I have shown this already. You see the BK knockout cannot do that. We can also quantify that. So you see less DNA outside of the cell. But these neutrophil extracellular traps are also functional. So we measure here, for example, bacterial killing. So the knockout, the wild-type neutrophils can kill, while the BK knockout cannot do it. And in these experiments, we measured also ROS, because we wanted to know, okay, we knew ROS is important for neutrophil extracellular trap formation, and you see there's no difference. So even the BK knockout neutrophils makes the same amount of ROS. So this is not the reason why they can't make ROS. And degranulation is also reduced in the BK knockout neutrophils. So next thing which I showed you, we need F-actin polymerization. So we checked F-actin polymerization, and you see wild-type cells can do it. The BK knockout cannot do it. And this we can also quantify. So basically what we learned is we have here a BK channel, which is important. We have normal F-actin polymerization. We have reduced F-actin polymerization. We have normal ROS levels. So what do we need for F-actin polymerization? We need ROS and we need ATP. So we thought maybe we have less ATP production by glycolysis in these BK knockout neutrophils. And this is indeed the case. So we have here mouse neutrophils. We compare BK knockout and BK wild type. So we have less ATP production. We have reduced glycolysis. And this is most likely owing to reduced glucose uptake of these cells. And we see similar results in humans. But in human neutrophils, we use a pharmacological approach because we don't have BK knockout neutrophils. And in this case, it's the same thing. When we use pharmacological inhibitors, here the PAX and the IBTX. So in both cases, we have less ATP production, we have reduced glycolysis, and we have reduced glucose uptake in these cells. So I mentioned already, so we moved a little bit also to the human neutrophils. So again, pharmacological approach, we see the same thing. When we reduce the function of the BK channel, we have less calcium influx in the cell, and we also have no neutrophil extracellular trap formation, no matter which of these inhibitors we use. Here in this case, in the untreated neutrophils, we have nice neutrophil extracellular DNA traps. So I mentioned already that we have reduced glucose uptake in cells which do not express the BK receptor, or we inhibit the BK receptor. So in this case, we analyzed a glucose transporter, so a molecule which brings glucose in the cell. And you see after stimulation of the neutrophils, we get immediate, within 1 minute, we get surface expression of this glucose transporter. So the glucose transporter is already preformed within neutrophils, translocates to the surface, and then glucose can go into the cell. So the BK knockout neutrophils, they cannot do it, so there's no glucose transporter on the surface, therefore no glucose can go into the cell. And the human system, same thing. So if we block the neutrophils with a BK inhibitor, we have no glucose transporter on the surface. So let me just summarize these findings. So BK is functionally expressed in neutrophils and regulate calcium influx. Lack of BK or its pharmacological inhibition leads to disruption of actin polymerization, degranulation, and neutrophil extracellular trap formation, but does not affect ROS activity. Ionomycin I haven't shown you, but it's also not so important at the moment. And BK regulates glucose transporter 1 translocation, which is required for glycolysis and ATP production. So the question is now, what is the associated calcium channel? So we know that potassium channels interact with calcium channels. So in order to find this out, we did a screening in neutrophils, and we checked the expression of different calcium ion channels. And you see both in the mouse as well as in human high expression of the ion channel TRPV2. So, and in this case now, we used the same approach, so I'm a bit quicker. So we used TRPV2 knockout mice. We isolated the neutrophils and we checked whether they make traps. And you see that these neutrophil extracellular traps cannot be done by neutrophils which do not express this calcium channel, and they have also reduced ATP production, and they have less glycolysis, which we measured here with lactate production. So is this whole thing important under in vivo conditions? So this is an infection model. So we infected the mice with Escherichia coli, and we observed how the Escherichia coli can be killed and whether these neutrophils can make neutrophil extracellular traps. Perhaps we check, we analyze, I show you first bacteria. You see normal neutrophils, they can kill all the bacteria, and in the BK knockout, you have lots of bacteria still in the organism. And the same is true with the TRPV2 knockout neutrophils. So wild type can nicely kill all the bacteria, but if this calcium channel is not in the neutrophils, they can't do it. And this is the case because the neutrophils cannot make DNA traps, and this is under in vivo conditions this time. So this is measured under in vivo conditions. Before I showed you everything under in vitro conditions. So these are the traps in vivo, and if the cells don't express the receptor, there's reduced DNA trap formation. So what I showed you today, and I'm a bit shorter, can't show you all the data, but I would like to give you the take-home message. So what we newly discovered is the expression of the BK channel and the TRPV2 channel in neutrophils. They interact together to get calcium in. So this calcium is bound on calmodulin, which activates the calcium chymotulin-mediated activated kinase 2, and this kinase phosphorylates the myosin light chain 2, and this kinase is important for F-actin polymerization. So at the same time, this kinase phosphorylates another molecule, and this is the glucose transporter 1, and this phosphorylation is required to get the transporter to the cell surface. This way then glucose can enter the cell, glycolysis can happen, and ATP is produced. So the work was done by Darko Stoikov. This was his project in collaboration with Kevin Oberson and Evelyn Kozlowski. And we had a nice collaboration with people who understand something about electrophysiology. And this is Peter Roth and Robert Lukowski from the University of Tübingen and Veit Flokerzi in Hamburg. Both are pharmacologists. Thank you very much for your attention.
Thank you very much. Awesome speech, and even we have time for a few questions.
Okay, maybe I start. What is the mechanism of exclusion of DNA and what kind
of? I forgot about this. So this is mitochondrial DNA, which is released. We have published it in several publications where we proved that this is mitochondrial DNA. We don't know. This is a short answer. We don't know which pores are required and which molecules are involved. We have done this with several knockout molecules or knockout neutrophils. We have tried to identify the mechanism also by morphological means. So at the moment we think that the granules, they fuse and make tunnels, and the tunnels open up on the cell surface, and that the DNA is first released into these granules and then released outside the cell, but we don't have enough good data to convince others.
Maybe a strange question, but could the neutrophil extracellular trap also play a role in exacerbation of an incident of the central nervous system? You see that the central nervous system, when there's damage, that there's a kind of enhancement of the damaged tissue, which is partly explained by activated neutrophils, but can the net also play a role? That are not bacteria, but what is your opinion?
Yeah, you're right, one can have an opinion about this, so we have also based on data, of course, but we believe that these neutrophil extracellular traps is a physiologic mechanism in which actually prevents tissue damage, because you localize the toxic molecules within this DNA network, and it's not as much distributed as without the DNA net. So it's very located in the toxic action in the tissues. So I think we prevent with this mechanism larger tissue damage.
A quick question. How long does this trap last?
It can last
before it disappears.
It can last several hours. So I have even, if I wouldn't be allowed, I have some movies. So it shows that the movies first show that the cells are viable because they are still moving. And often it's the case you activate the release of these neutrophils, and later these neutrophils die, because we show here physiological activation. Cells don't die, but some people use PMA, for example, and then the cells die, say, within 2 hours. And PMA stimulates also neutrophil extracellular traps very fast, say, within 1 minute, and then after 2 hours, the cell dies. And then you see both. First, you see the neutrophil extracellular traps, and 2 hours later, the nuclear DNA comes out, and the neutrophil extracellular traps are still there in spite that the cell dies. So that means the traps can stay over several hours for sure.
This could be very dangerous, no? It could cause autoimmune
Yes,
reaction.
this is true. I mean, we talked before about tissue damage, direct tissue damage by granule proteins, but of course these cationic proteins together with the DNA could stimulate also toll-like receptors and then stimulate through dendritic cells' immune system. So there's a potential of autoimmune reactions.
Oh, very interesting. I have a question about the mitochondria who have released the DNA outside. They remain inside the neutrophil.
Yes.
What happens to them? Are they removed by autophagy or I don't know?
Well, we didn't study autophagy, but the mitochondria, at least the majority of the mitochondria, they stay within the cells. And they still have also some mitochondrial DNA, but less, of course, because some is released. We try to quantify that with different techniques. We get different results. So, for example, when we use PCR, we would say 20% of the mitochondrial DNA is released in average of the cell. If we do it by staining techniques, we have, but I think this is a sensitivity issue, then we have the impression that they release more. So that maybe 50% of the DNA is released if you quantify it with color. Yeah. So, and what was your question? Yeah, and the neutrophils, I think that they don't need the mitochondrial DNA as a cell. So therefore they can get it out and they are still functional normal.
Because neutrophils are not long-lasting cells, so they live 24 hours or
It
something like that.
depends.
So probably they don't need to synthesize mitochondrial proteins.
In isolated neutrophils in culture, they live 1 to 2 days, but in tissue they can live longer, up to a week.
Okay, thank you very much. And I invite next speaker and our partner, Dr. Mohan from BGI Research, Institute of Digital Earth, and his research focus on microbiome on extreme environment.
Okay, so good afternoon, everyone. It's my pleasure to stand here and introduce our work on studies of microbes in the extreme environments. I do understand that most of the audience today are not working in the field of us. So I will try to keep my presentation quite simple. Okay, so just a brief introduction of myself. I have been working in BGI research for about 10 years or a little bit longer. In the first 5 years, I basically working in human gut microbiome research. And after that, I moved my focuses onto the extreme environments. So how it happens? It is because that while some flagship animals, as polar bears, penguins, and snow leopards, these amazing animals living in some harsh environments. But most of the life inhabit in such extreme environments are basically microorganisms. So the problem is that what is living there and how can they adapt this kind of harsh environments, and what's the relationship between these life living in such remote and harsh environments and human beings. To answer these questions, we and our collaborators collected more than 10,000 samples from polar regions and hydro trenches, as well as the high altitude plateaus during the last 2 decades. And by sequencing all of these, these treasure samples, we have unveiled the life living in deep sea, in polar areas, and also in glaciers and high altitude snow mountains. So today let's start from deep sea, yeah. Okay, so the Mariana Trench Environment and Ecology Research Project, short as MIR project. This is the biggest oceanic microbiology sequencing program up to date. We have generated 92 terabases of sequencing data for microorganisms and 250 terabases for animals. As for these microbes, almost 90% of these prokaryotes are unreported, and more than 99% of the viruses have not been included in previous studies. By conducting this project, we unveiled diverse adaptive strategies of the microorganisms, and also showed the power of the new paradigm of sequencing technology. We collected 1,642 segment samples from the Mariana Trench, Yap Trench, and Philippine Basin near these 2 trenches. Metagenomic sequencing has generated, or say recovered, more than 7,000 of species-level genome bins from the sequencing data, and only 7.2% of these species have been reported in previous metagenomic studies of the Mariana Trench. This showed that high depth sequencing for the extreme environment samples can lead us to a more comprehensive understanding of these uncultured microbes. And by analyzing the annotated functional genes, we found that the bacteria and archaea living in these deep sea environments has 2 different adaptation strategies. One of them is streamlined strategy, which means the microbe will streamline their genome and reduce their capacity of metabolism to keep the energy consumption as low as possible to living in this oligotrophic environment. But on the other hand, some of the species chose to live in a versatile way. They have a relatively larger genome size comparing to their relatives living in surface ocean, and can therefore utilize more compounds to live. Not only prokaryotes, viruses are also contribute in the extreme environments. Actually, we have identified more than 1.3 billion million of viral contexts, and these contexts can be clustered into more than 800,000 viral genomes. Of course, most majority of these viruses has not been reported. Actually, these viruses are not just pathogens of the prokaryotes. Many of the actually metabolic genes harbored in these viral genomes. Most of these genes are related to the carbon metabolism and compound transportation between the different cells. So actually they are not just harmful pathogens for lives living in extreme environments. They also help them to adapt to the environment and to live there better. After the deep sea studies, let's ascend a little bit to the top of the roof. We also studied the environmental microbiome of the Himalayas. Indeed, we collected about 40 samples from the mountain Chumlama and mountain Chaoyu during the last 2 years. It is really hard because you need to climb to the peak, and otherwise you cannot have the samples. So by sequencing these samples, we recovered more than 1,000 species, microbial species living there, and we found no dominant phyla. It is very different from the previous reports of the glacier microbiome. We don't really know what's the differences, but one thing to mention is that there are some marine-related taxa can be found on the very high region of the mountains. It can be a consequence of the air and rain, but no direct evidence has been found. These microorganisms living in such high altitude showed distinct genetic characteristics. Here I would like to mention two of them. The first one is that while the generic metabolism potential has been reduced, most of the species enriched in the most high regions showed an enhanced potential in reducing nitric oxide. So we checked for the denitrification pathway and found that near half of the energy released in this last step of reduction. So this can be a clue to understand how these microbes living in this kind of energy restricted areas. And also, we found that these high altitude enriched species showed exclusive preference in cytochrome B deoxidase comparing to the cytochrome C oxidase. This is because that cytochrome B deoxidase has much more stronger how do you say? A preference or affinity to oxygen. So a preference in cytochrome B deoxidase can help these organisms to adapt to the high altitude environment. So yes, they are very interesting, but what's the relationship between these microorganisms living in high altitude with us, human beings? Actually, we also collected some samples from the northeast part of the Qinghai-Tibet plateau to check the relationship between microbiome with the global climate change. And after recovering the microbial species from the 6 drilling cores collected, we found that the taxonomic richness showed a negative relationship between the with the permafrost degradation. So suggesting that during the change of global climate, the microbial diversity will decrease. And within this period, we also found that the microorganisms which can generate nitric oxide showed an increase with the degradation of permafrost, suggesting that there can be a kind of amplified feedback between the global climate change and the shifting environmental microbiome. Aside from these indirect relations between extreme environments, microbiome, and us, there are also some more direct connections. The first one is that we have identified different kinds of antimicrobial compounds from these microorganisms, including antimicrobial peptides and novel kind of traditional antibiotics, which can inhibit the growth of MRSA. And we also found some interesting enzymes which may be useful for future biotechnology and environmental governance. Utilizing these and some of these enzymes we found, we also launched, actually this is not launched by BGI Research, but a commercial company of BGI Group, MGI. we launched our own long-read sequencing platform using nanopore protein and motor protein identified from deep-sea microorganisms, which enabled very high throughput and finite sequencing results. So overall, our studies, our research is trying to face the global challenges, including climate change and the desires in novel drugs. We would like to establish kind of microbial resource banks, and also to review the microbial diversity and their adaptation mechanisms to the extreme environments. And also, we will try our best to bring some affordable and advanced methodology based on our understandings of these extreme environmental microorganisms. Okay, I think this is just all for today, and thanks for all of our collaborators and all of your, thank you.
Thank you very much. It's a fantastic speech, and it's a really important speech for our group because of inconception of One Health. It's really important to understand the connection between human health and environmental microbiome. So because of tight time, we leave in the end of our section, the time for discussion section, and now we can continue. Welcome, please. And last year, we win international grant from Open Biome, where participated 70 countries, it seems to me, and I would like to invite our partners from this grant, Dr. I'm sorry, just a minute, please. Dr. Lucas Matilha Silva from Institute for Clinical and Molecular Biology, Institute of Experimental Medicine, Kiel University, German.
Hi, thank you. Thank you, Professor Amagu, for the kind invitation and the introduction. Yeah, I'm very excited to be here in Kazakhstan. It's my first time, and I'm also happy to talk a bit about our work. That is also in collaboration here with the university. So I will talk today about, I'll talk today about the effects of the relationship of industrialization on the gut microbiome. Right, so as Mohan mentioned, I reckon also that the audience here is very broad. So just talk slightly broadly about human microbiome. So it's been a hot topic in the last 10, 15 years. So basically what I call human, or what we define as human microbiome, is the set or the group of microorganisms that live in the human. So colonize a human and can colonize different surfaces like mouth, skin, urogenital tract, and digestive tract. And this could be bacteria, virus, and fungi. And just to give a perspective of the dimensions of the human microbiome, and now I'm going to talk specifically about the gut microbiome, it's estimated that for each nucleated cell in the human body, there are approximately 10 microbial cells in the gut. And together, they sum up to, collectively, to more than 3,000 microbial species, which can be estimated to 200 milligrams of wet weight. So this is estimation for male, 70 kilograms. So it's quite visible if you put all together. And there are several factors that shape the gut microbiome, such as age, lifestyle, medicaments, geographic location, depending on the country, disease, gender, probiotics, so if you ingest microbes as well, or diet. And all of this research and knowledge most of it have been produced based on data, based on human, European, and North American samples. And so, as I mentioned, in the last 10, 15 years, there was a peak in this, you can see the amount of samples that were cumulatively submitted to public repositories, and most of the data is dominated by European and North America, and this actually represents a very small part of the world population. So that means that the knowledge that we have right now is biased towards these populations and this lifestyle, genetic background, and so on. So in order to fill the gap, to better understand the human microbiome and all its factors shaping it, Mathieu Depoyer and Mathieu Grossin, they are the PIs, so my bosses actually, and they are the PIs of Global Microbiome Conservancy, and which aims to fill this gap by sampling across the globe in different lifestyle backgrounds. So for instance, we are very happy with our collaborators here in Kazakhstan as well, Professor Amagul. As you can see, within each country, we took care of sampling from locations with a different lifestyle gradient. So here, for example, in Kazakhstan, there were samples from in Astana, spanning to West Kazakhstan. And these are the collaborators and the people responsible for collecting the samples. And the samples from Kazakhstan arrived this year, and they are still being processed. So the results that I'm going to talk about it here, they are not, the samples were collected here are not part of it, but they will be incorporated as the research goes. So as I mentioned, the Global Microbiome Conservancy aims to include a diverse spectrum of individuals. So in total, the results I'm going to talk to you about includes 1,000 individuals, and for them, we collected extensive lifestyle metadata as well as diet. We also collected saliva samples. By we, I mean collaborators of the Conservancy. And the saliva samples were collected for human genotyping. Fecal samples were collected for metagenomics, which is basically retrieving the genomes of the microbes present in the fecal samples. We also did anaerobic culturing for isolating and sequencing or genome, producing the genomes of isolates, as well as biomarkers, such as IgA, calprotectin, for instance, which is a biomarker of gut inflammation. What you see here, it's a PCA, so it's a reduction of the millions of SNPs that are present in the 1,000 Genomes Project. So all of these millions of variants, genotype variants, they are reduced in 2 dimensions here. And you can see in green are the samples that were collected from the GMBC. And you can see that it spans quite well from African populations to American and East Asian. So we collect a broad spectrum of individuals here. We also, same idea here, showing a PCA now for the lifestyle. So we collected several information about their lifestyle, reduced this information in 2 dimensions, and this is what I'm showing here. And you can see that the graph, there are clusters based on country. But when you lay this over industrialization, you actually can see that in the first dimension, there was a quite distinguished here of the gradient from non-industrialized populations to industrialized populations. And when we look at the phylogenetic diversity of the bacteria or the microbes from the gut microbiome, there is a negative correlation of according to the PC1, which captures the industrialization. So when we lay this over the industrialization, we can see that there was a decrease in microbial diversity in industrialized societies. When we look then in a more systemic approach, where we did co-abundance networks. So here are the networks where each microbe, which are represented here by the dots, when they are co-abundant, whether positively correlated or negatively correlated, they are connected by a line here. And what you can see is that there was much more connections in the non-industrialized gut microbiomes in relation to the industrialized. So when you translate that, it means that there was a decrease in microbiome stability in industrialized societies. We also went further and we look at which microbes were actually different between industrialized and non-industrialized lifestyle. And in the graph above, you can see the individual results. And here in the graphs below, we see the summary, where we can see that the gut microbiomes of non-industrialized population enriched in Prevotella, for instance, bacteria, while the industrialized are enriched in Bacteroidetes. I mean, this is just the tip of the graph. There is a long tail there, but these are the main genus that were found. We repeat a similar analysis, and here I'm showing to you the functions, here summarized by KOs, which is KEGG ortholog. So basically, each KO represents one function, one microbial function, and these are the spectrum of the functions that were found associated within, considering the countries and the lifestyle and the genetic background that were associated either with non-industrialized populations or with industrialized populations. And we look at further, not just in individual functions, but also in modules and pathways. And we found a couple of pathways that were associated with these different lifestyles. And here I'm just showing one of them, which I found quite interesting, which is the biosynthesis of vitamin B12. So we found that microbiomes from industrialized societies, they were enriched in biosynthesis of B12. And this was based on the metagenome data. We then confirmed this data based on gene-wide associations, based on isolates, genomes of isolates. So just to illustrate all these words that I just said, what you can see here in my right side is actually the genome of Bacteroides uniformis, and this is an isolate. So for this we have in culture, so we have a good, the complete genome of it, and for each single nucleotide polymorphism, we did associations, whether they are associated with lifestyle or not, and we found that these are what were found in this particular genome, and this includes one protein, so one variant of a protein that were associated with lifestyle. This is just one example. We also found variants in other proteins also related with the biosynthesis of vitamin B12. So this is quite interesting because we arrived in the same place looking on the metagenome presence of and abundance of functions, as well as the adaptative variants of proteins in the genomes of bacteria. So with that, I would like to conclude that the GMBC provides a robust framework and a resource to understand the impact of human lifestyle on the gut microbiome. Microbial composition and functions are impacted by industrialization of lifestyle, so there's a really big difference between what you found in terms of functions and which kind of microbes are found, whether in respect to industrialization. And there is a need for representation of cohorts from non-industrialized societies for a comprehensive understanding of gut microbiomes. Again, this is a target approach trying to fulfill this gap, but they are just 1,000 samples, and these are actually quite It's quite a big effort because it involves many collaborators and participants across the globe, but it also shows that there is a long path to be walked. With that, I would like to thank all GMBC collaborators, including from here, from the university, as well as the participants. So we will also try to give something back. We have, for instance, produced reports about the microbiomes for the participants that want to know more about the data that they provided. And also the lab members of Poirier and Grossan Labs, which are actually the people that were behind the graphs here shown. Thank you very much for your attention.
Okay, thank you, Lucas. Because we are running out of time, so let's discuss after the conference. And now let's welcome our next speaker, Professor Mauro. She is a professor in Medical University Astana and the chair of the Committee on Academic Affairs of the Academic Council of Astana Medical University. Welcome.
Good evening, ladies and gentlemen. Announcement, because of the speech will be in Russian, would you please use the earphone?
Добрый вечер, уважаемые дамы и господа. Я приветствую вас. Сори, что я меняю формат на русском языке, но тема моя очень будет интересная для многих присутствующих. Мы впервые доложим результаты наших казахстанских исследований совместно с Назарбаевым университетом. Итак, мы поговорим сегодня о микробиоме новорожденного и становлении иммунной толерантности. И, конечно, на сегодняшний день уже абсолютно известно, что ключевой фактор формирования иммунной системы именно микробиом новорожденного. В постнатальном раннем периоде, именно в постнатальном раннем периоде формируется уже иммунная толерантность, которая определяет риск развития аллергических, аутоиммунных заболеваний. И последние педиатрические отчеты, последние педиатрические доступные отчеты, которые мы имеем, позволяют сделать вывод, что состав материнской микробиоты оказывает большое влияние на неонатальный иммунитет и риск развития аллергии. Именно первые месяцы жизни являются особенными, и любые нарушения, связанные с кесаревым сечением, антибиотиком, отсутствие грудного вскармливания, конечно же, сдвигают риск толерантности в сторону гиперреактивности и недостаточной защиты. Последние годы особенно отмечены всплеском научных исследований в плане доказательств исследований микробиома, и мы видим систематические большие обзоры, мы видим серьезные новые математические и мульти-омикс модели, о которых только что сейчас указывал наш предыдущий спикер, которые показывают о том, что возможно обучение именно иммунитетом. Это делает нашу проблему наиболее актуальной для профилактики и клинической практики. И вы видите количество публикаций, более 200 за последние только годы, которые объясняют механизмы микробиома. Основные понятия в отношении микробиома, конечно, известны, но я хотела бы обратить ваше внимание на критическое окно. Это период взаимодействия между микробиомом и иммунной системой, которое наиболее такое пластичное. Особенно первые месяцы, 2-3 месяца жизни до 2-3 лет, особенно после отлучения от груди. Конечно, в этот период очень сильно меняется у нас уже иммунитет. Что касается состава микробиота, то тоже микробиома, многочисленные исследования показали, что мы знаем на сегодняшний день, имеем понимание, какие основные группы бактерий присутствуют в микробиоме кишечника. Вы видите, у детей грудного возраста доминируют, особенно при грудном вскармливании, это актинобактериям, фумигитус участвует в ферментации и регуляции, протеобактерии бактерия встречается прямо в первые недели жизни, постепенно потом уменьшается, и бактероиды сформируются уже чуть позже, и особенно после введения прикорма. Ну, существуют и дополнительные компоненты, которые, безусловно, влияют и модулируют состав микробиома. И как уже оказалось очевидным, на сегодняшний день источники формирования микробиома известны у новорожденных. Вы видите, это, как правило, материнский микробиом. Это кожа, кишечник, метод родоразрешения, антибиотики, антибиотики у матерей и новорожденных, которые использовали пациенты, и как влияет тип питания, как оказывает влияние окружающая среда на течение формирования иммунной толерантности. И вот обращаю ваше внимание, что грудное молоко содержит пребиотики, бактерии, иммунные компоненты, именно иммуноглобулин А, который способствует именно формированию и развитию иммунной толерантности. Что касается механизмов, основные, конечно, механизмы уже известны в формировании толерантности, и они напрямую связаны прежде всего со стимуляцией врожденного иммунитета, именно через взаимодействие ТОЛ-рецептора, рецепторов дендритных клеток на макрофагах, регуляция регуляторных Т-клеток, которые участвуют непосредственно и стимулируют развитие Т-регуляторных клеток и тормозят или активизируют именно иммунную систему. Укрепление барьерной функции, метаболиты микроорганизмов – это те основные точки, которые на сегодняшний день именно способствуют предотвращению миграции бактерий. Недавние исследования, которые последние годы особенно побудили нас к проведению исследований, это, наверное, результаты исследований кафедры экологии и эволюционной биологии Принстонского университета. Типиколи и его коллеги создали первую математическую модель раннего возраста обучения иммунной системы под влиянием микробиома материнской среды и факторов. Доказана была ключевая роль грудного вскармливания и иммуноглобулина А в формировании иммунной толерантности. Было показано, что кал младенца может служить предиктором будущих иммунных траекторий. Другие исследования авторов тоже продемонстрировали достаточно интересные данные. Вот метаанализ проведенный был, который включал 74 исследования. Однозначно было сказано, что кесарево сечение, антибиотики, стресс повышают риски в развитии аллергических заболеваний. Материнский дисбиоз у животных ухудшает функцию плаценты, снижает активность натуральных клеток и нарушает ангиогенез, влияя на обучение иммунитета. Антибиотики новорожденных связаны со снижением антителообразования, с плохим иммунным ответом на вакцины и, конечно же, со снижением бифидобактерий. И исследования тоже китайских ученых по поводу влияния кесарево сечения и уровня липополисахаридов, тоже очень интересное исследование, и прежде всего было указано, что изменялся состав, изменялась экспрессия ТОЛ-рецепторов и формировался противовоспалительный статус у этих пациентов. Факторы, которые были обозначены после исследования этих всех публикаций, конечно, побудили нас к исследованию, и мы проанализировали литературу, и действительно были обозначены факторы, нарушающие полностью иммунную толерантность. И вот здесь в данном случае это кесарево сечение, избыточное или неправильное применение антибиотиков у матерей и детей, это плохой ответ, неполное грудное вскармливание или ранняя замена смеси на пребиотики, плохое питание матери, низкое потребление клетчатки, низкое содержание омега-3, плохое разнообразие, стресс и или ожирения. И, конечно, среда слишком стерильная, которая исключает антигенную нагрузку с природной микрофлорой, тоже способствует снижению иммунологического развития. И какие последствия нарушенной толерантности мы можем увидеть? Последствия. Повышенный риск аллергических заболеваний, именно астмы и атопического дерматита. И мы видим рост распространенности аллергических заболеваний у детей в последние 30 лет. Плохой ответ на вакцину, снижение титров антител у новорожденных детей, которые получали антибиотики. Вы видите, вот пероральные вакцины, ротавирус, полиовирус и другие вакцины, в сравнении с контролем, конечно, титр антител был ниже. И, наконец, еще один момент, возможно увеличение риска аутоиммунных заболеваний на фоне дисбиоза, раннего дисбиоза. Именно рост аутоиммунных заболеваний и воспалительных заболеваний кишечника, языка у подростков в течение 30 лет был увеличен в несколько раз. И когда проанализировали данные, было отмечено, что эти дети имели признаки дисбиоза, причем раннего. И, наконец, долгосрочные изменения в иммунной регуляции, которые способствуют снижению устойчивости к патогенам, усилением воспалительных процессов, тоже подтверждаются повышенными уровнями провоспалительных цитокинов у детей с ранним дисбиозом. Казалось бы, имея такой арсенал уже данных, огромное количество публикаций, в то же время остаются ряд открытые вопросы в отношении именно влияния географических, этнических, социальных факторов, различий между популяциями, долгосрочные исследования, механизмы на молекулярном уровне, нужны ли пробиотики или нет, какая доза, в какой период, кратность применения, какие штаммы безопасны, какие эффективны, это на сегодняшний день, конечно, еще открытые вопросы. И пытаясь как бы ответить, попытка ответить на эти вопросы побудила нас именно впервые в Республике Казахстан начать изучение микробиома матери и ребенка. В рамках совместного проекта программирования здоровья и эволюции микробиома младенца с Назарбаевым университетом мы, вот группа авторов, которые здесь присутствуют, я их всех рада видеть и приветствую. Уже выполнена была магистерская, докторская диссертация. Это только начало большого пути. Но в то же время, конечно, нам удалось уже увидеть взаимосвязи между младенцами и матерями, их матерями. Первое исследование – это фрагмент, небольшой фрагмент, который мы вам представляем. В исследование были включены 253 матери, 253 младенца. Это городские дети с 15 поликлиник. Исследование проводилось на биобанке младенцев, лаборатории микробиома человека и долголетия Назарбаева университета, Национальной лаборатории Астаны. Критерии включения – это матери и дети без патологии от рождения до 12 месяцев. Критерии исключения – это дети с врожденной патологией, недоношенные дети и не имеющие добровольного согласия. Методы исследования включали анамнестические данные, антропометрические данные, метагеномные исследования. И вы видите в динамике при рождении 1, 3, 6, 12 месяц исследовались анализы кала, слизистых у детей, и микробиом матери изучался через месяц после рождения. Были изучены ряд общих детерминант здоровья, это возраст мамы, средние, видите, 30, показатели, акушерский анамнез, паритет родов, количество детей, интервал между родами, метод разрешения 81% это естественные роды, срок гестации соответствовал однозначно нормальным показателям, способ кормления. Социальные детерминанты – это виды трудовой деятельности во время беременности, вредные привычки, употребление алкоголя и курения, тип используемого транспорта, частота посещения общественных мест, условия проживания, то есть жили в квартире, вместе с родственниками, отдельно. Такие нюансы тоже учитывались. Характеристика питания матерей, употребление молочных, овощных продуктов, мясных продуктов, употребление, то есть в целом оценивали рациональное питание беременных женщин. Патологические состояния были анализированы. Патологические состояния были также проанализированы. И среди них отмечалось, конечно, это гипотиреоз, аллергические заболевания, преэклампсия, острые заболевания, у каждой третьей были ОРВИ, и в целом хронические заболевания составляли до 80%, анемии беременных составили 88%. Кроме того, впервые нами мы проанализировали прием антибиотиков, антимикробных препаратов во время беременности, прием лакто- и бифидобактерий, то есть пробиотиков. И видите, какой небольшой, совершенно незначительный процент был. Показатели здоровья детей были достаточно позитивные, уважаемые коллеги, и вакцинирующий этап был достаточно такой высокий, но в данном случае исследование было продолжено, и, конечно же, в первую очередь был проведен анализ фекальной микробиоты с помощью шангун-секвенирования, и использовался именно метод профилирования микробного сообщества, который позволял около 44 миллионов прочтений каждого образца. Результаты этого анализа показали, что бифидобактерии, бактероидес и шерихи являются преобладающими бактериальными родами фекальной микробиоты в раннем младенчестве. Были обнаружены с первого месяца жизни изменения в микробиоме, которые были связаны с младенческими коликами. Было обнаружено преобладание таксонов, принадлежащих к классу Кластридия, у детей в 4 раза больше с коликами, вместо детей, у которых не было младенческих колик. У детей без колик преобладали бифидобактерии в значительном проявлении в сравнении с группой без колик. Кроме того, было изучено альфа-разнообразие, то есть количественный состав, был изучен анализ бета-разнообразия между точками, то есть сравнительный анализ между материнским геномом и геномом ребенка, также анализ расширенных графиков погрешностей, который выявил 47 значительно различающихся метаболических путей, связано с таксонометрическими нарушениями в микробном сообществе. И рандом-форест анализ, который тоже показал нам различия в этих группах. И результаты нашего исследования, конечно, побудили нас в дальнейшем продолжить это исследование. В дальнейшем хотелось бы посмотреть, как развиваются дети и в 2, и в 3 года, как изменяется состав микробиоты, и как будет влиять дальше социализация, питание и образ жизни у этих детей. Что касается выводов, микробиом новорожденного, конечно, это главный фактор формирования иммунной толерантности. И на сегодняшний день необходимо продолжить исследования, особенно исследования рандомизированные, контролируемые, с долгосрочным наблюдением, потому что действительно есть такая необходимость, возможна интервенция и профилактика для поддержки здорового микробиома. На сегодняшний день это реалии. Благодарю вас за внимание. Спасибо. Thank you very much.
Okay, thank you, Marina. Now let's invite our next speaker, Professor Natalia Latpova. She's also working in the Medical University Astana, and director of the Research Institute of Respiratory Medicine. Let's welcome her to introduce the burden of lung diseases in Kazakhstan. Welcome.
Dear colleagues, I'm pleased to welcome you all here for this conference. I would like to thank committee members to make this conference possible. Sorry. And I appreciate the possibility to be and speak here. Pardon. Some words about objectives of my topic. You might notice that my report does not fit the scientific agenda of, sorry, what does it mean? Of this particular symposium, but it entire refers to practice and scientific challenges in respiratory diseases in Kazakhstan. I would like to begin with statistics data. Our official statistics says that respiratory disease ranked first in morbidity and third in mortality just after oncology in 2024. Due to the largest study named the Global Burden of Disease Study, published in 2023, Kazakhstan figures correlate with worldwide statistics. Due to this study, Kazakhstan is in the red zone of mortality and has a high daily index. This pie chart demonstrates figures of mortality due to respiratory disease in Kazakhstan. As you can see, the COPD is the leading cause. Oh, I don't know what this. Leading cause of mortality, and pneumonia comes next, and both COPD and pneumonia make approximately 25% of mortality. Major burden of respiratory disease in Kazakhstan can be grouped in four categories: pneumonia, chronic obstructive pulmonary disease, bronchial asthma, and interstitial lung disease. Pneumonia is in the top 10 disease by frequency of hospitalization in Kazakhstan and has a highest inpatient mortality among therapeutic profiles. COPD and bronchial asthma has an increasing trend in Kazakhstan, and the problem of interstitial lung disease has new level after pandemic in Kazakhstan. Before we consider tendency of viral respiratory tract infection in Kazakhstan, let me demonstrate worldwide experience. The number of studies find out the three key tendencies in respiratory infections. The first one, the frequency of respiratory infection has risen. Seasonal patterns has changed. The co-infections with multiple viruses has considerably increased. A recent study find out the same tendency of pneumonia. For example, this one study shows that pneumonia increase causes by multiple bacteria in viruses. And considering Kazakhstan, we can see the same tendency, dramatic increase of incidence of respiratory infections. And we can see mortality caused by pneumonia increased by 2 times. Due to pneumonia in Kazakhstan, we can see 30% increase
30% increase of hospitalization due to pneumonia in 2024. In this year, the total number of hospitalization was over 200,000, maintaining the high level in summer period. The global burden of respiratory disease largely related to non-communicable disease. World Health Organization puts chronic respiratory disease in the top 4 global problem, along with cancer, diabetes, heart disease, and stroke. And COPD and bronchial asthma demonstrate increased trends in Kazakhstan, and mortality rate remains high, primarily COPD associated with respiratory failure. Interstitial lung disease, there is a problem in Kazakhstan, and should be noted that approach to manage interstitial lung disease is changed recently due to pandemic period. Diagnostic of interstitial lung disease has been improved due to frequent use, computer tomography, and implementation of national registry of ILD in Kazakhstan, which helps to monitor patients more effectively. Moving on to economic issues, take COPD as an example. Annual cost of I represents in billions. This graph demonstrates expenses on outpatient and inpatient care, and we can see huge expenses. So lung disease affects the system of healthcare in Kazakhstan seriously. The following slide demonstrates key healthcare strategy to reduce mortality of respiratory disease in Kazakhstan, mainly based on prevention, early stage of diagnostics, outpatient management, monitoring patients with high-risk exacerbation and mortality. Scientific and practical activities in the field of respiratory disease in Kazakhstan is still developing and being supported. Research institutes and scientific center, of course, Nazarbayev University Research Institute of Respiratory Medicine, and National Scientific Medical Center, and other universities and institutions. It's important that collaboration has significantly strengthened for last years, and our cooperation with Nazarbayev University is a good example for us. And the last but not the least, our major goal focused on translational medicine, so-called from bench to bedside. And I would like to point out some areas of this, such as early diagnostic and prevention, personalized medicine and phenotyping, and of course development of new therapeutic targets. I hope that we are moving in the right direction, and that's all. Thank you for your attention and staying here until the end of this symposium. Thank you.
Okay, thank you, Natalia. And now let's welcome our last talk presented by Professor Amago Kushkulova director of the MICROBIOME Research Center.
Okay, thank you very much. I recognize we are all rather tight, and so I will keep my presentation brief to leaving time for discussion and some question, and ensuring that we don't miss welcome reception dinner. So my research focused on, of course, the primary focus is to identify new novel biomarkers and therapeutic targets for preventive progression of diseases in this time atherosclerosis. So the Shannon disease, the alpha diversity, we observe that in atherosclerosis group, in comparing with healthy control, we have a significant decrease. That means that microbial dysbiosis is characteristics of atherosclerosis. And the next image, we can see the beta diversity PCA analysis show us that we have a significant pronounced separation between the two groups, healthy control and atherosclerosis, that confirm that the decreasing of microbial diversity and characteristics of atherosclerosis dysbiosis. This, we have another group, not only atherosclerosis, but the patients with low cardiovascular disease, and compared in two metrics, Bray, Curtis, and Jacquard, we can observe that we can see the pronounced separation between healthy control and low cardiovascular disease microbiome, but of course, in comparing with previous atherosclerosis, it's less pronounced. But it means that the shift, that the changes of microbiome are much more earlier than in disease. And the analysis show us the progression of microbial dysbiosis from low cardiovascular to atherosclerosis. In atherosclerosis group, we can see the high level of opportunistic flora, for example, Clostridiales. Clostridiales, it's a tax, is a source of LPS and is a source of imidazole propionate. It's a novel cholesterol-independent biomarker for atherosclerosis. And in low cardiovascular disease microbiome, we can observe the compensatory increase of Faecalibacterium prausnitzii. It's a beneficial microbe, but in that case, we can see that our microbiome can try to protect our organism from progression of the disease. And Faecalibacterium prausnitzii, it's a butyrate-producing bacteria that lead to anti-inflammatory effect and lead to decrease in the synthesis trimethyl amino acid. In our patients, we can see the negative correlation with ApoB, like a protective effect of microbiome. And in this slide, we can see that our microbiome produce much more harmful metabolites, inflammatory metabolites, and fewer beneficial fatty acids. And we can, additionally, we can observe that our metabolism changed critically in comparing with healthy control, and lead in the markers of total inflammation process in our organism. So additionally, we stratified our patients, atherosclerosis patients, into group obese with and non-obese, and compare. And on the first image, we can see the PCA analysis show us the significant separation between two groups, and due to the higher genus of Prevotella. It's 7-fold than in healthy group. Prevotella is a producer of LPS like a Clostridiales. It's a gram-negative bacteria, and it leads to inflammatory process. So here we can see the positive correlation of genus Prevotella with branched chain fatty acids with lysine and isoleucine that showed that disrupted mTOR inflammatory cascade way. So I tried to be short, so just the mechanism, improper diet, or maybe something other stress for organism lead to gut dysbiosis. That means that decreased beneficial bacteria like lactobacillus, bifidobacteria, Faecalibacterium prausnitzii, and the higher opportunistic flora like Prevotella or Clostridialis. And of course, we observe that decreasing the butyrate produces bacteria. It means that it's not simple dysbiosis. The Prevotella and Clostridialis is a source of, due to some physiological process of this bacteria, we observe the high concentration of LPS inside intestine. And due to decreasing butyrate producing bacteria, the tight junctions, the proteins between that connect to epitheliocytes, they became weaker. So because of butyrate is the only source of energy for goblet cells and for epitheliocytes. And because of we additionally observed that mucin degradation process in previous slide. So we have a translocation process. So LPS goes to systemic blood. It means that, and it uses the cascade of toll-like receptor 4, nuclear factor kappa, and inflammasome pathway that leads to classical atherosclerosis pathway. So we performed this study in collaboration with Heart Center, and here our leading physicians. Heart Center is under the umbrella of Nazarbayev University, and additionally they are all unit, the entire physicians of Kazakhstan specializing in atherosclerosis, and it's our team, mostly young researchers in their career. So thank you very much. The shortest speech today, it seems to me. So we have enough time for questions for all speakers of this section.
Just interesting, curious question regarding this microbiome of infants and mothers. Professor Marina maybe can answer, or if not, maybe Almagul. Because it's interesting, sometimes it's modern to have a birth in water pool. Is this influence on microbiome of infant or not? Because some of these factors was influencing, it was mother microbiome. Who will answer?
Я переведу. Я говорю, иногда модно женщины рожают в этих, как его, бассейнах. И говорят, что это типа среда такая же, жидкость, там, вода, то все. Насколько это влияет на микробиом младенцев? Есть ли такие исследования? Или
можно еще раз вопрос, да?
Влияние родов в бассейне? Есть ли такие исследования или какое может быть влияние на формирование микробиома у младенцев?
Ну, исследований очень мало в этом направлении. Спасибо за вопрос, да. Мне встречались в литературе исследования, где говорилось, конечно, все зависит от качества, да, бассейна, от воды, от той среды, которая находится. Но однозначно некоторые бассейны действительно используют антисептики в избытке, что влияет точно так же, как и антибиотики, любые антисептические средства. А другие, наоборот, не выдерживают никаких санитарных норм, да, что еще больше способствует колонизации. Поэтому мы сталкивались в нашей стране с теми вот моментами, и, конечно, однозначно приветствовать этот вид не рекомендуют. Спасибо. Физиологические.
Но ваши 253, они не вошли?
Нет.
Хорошо, спасибо.
Any question else to all speakers? Okay, Artur.
Okay, My question to the Lucas. I saw the very interesting detail in your presentation. I don't know, can you show it or no? About the distribution between industrialized and non-industrialized areas, and I saw that there was talk about Prevotella and bacteroids, and the interesting thing that we discussed it a lot. For example, our laboratory based in Astana, and Astana is one of the industrialized cities in Kazakhstan. So the answer for us that half of our population have dominated Prevotella. And this is very interesting topic because when we had collaboration with our USA colleagues, we saw this a big difference in Prevotella, that half of our population had dominated Prevotella, and in USA, none of them had Prevotella or it was negligible. And when we researched it, we came to the conclusion that according to the researches that were conducted in Madagascar, India, Japan, USA, and some European cities, that is more geographical marker, and it's very, very dependent on diet. So the difference was in Western diet and in Asian diet. So my question is how your industrialized and non-industrialized areas are distributed on geography. Is it that most of the industrialized cities that you have is in USA and Europe, and non-industrialized is near Asia or Africa?
Yeah, thank you. This is a very interesting question. I'm very much looking forward for the data that will be generated here from Kazakhstan, because I think this will be really the data that could answer these kind of questions. What I can say from the results that we produced is that they were corrected for diet, lifestyle, and genetics. So the information that we unfortunately, I'm not able to come back. Yeah, one more, maybe one more. Yes. You can see that we made two models, and one was lifestyle alone, so basically having industrialized versus non-industrialized, and the other one was lifestyle plus confounders. And these plus confounders, of course, you can see that most of the diamond symbols are reduced, effect size, so, but many of the results still stand even after including the confounders in the models. So I think to answer shortly your question, I think, yeah, to recap, one, this result, we are relatively confident that is independent of lifestyles and genetics and other confounders. Two, I think the sampling design that was done here in Kazakhstan, including Astana and other sampling sites, will be really good to target that, because yes, there is one country is a big effect on how the microbiome looks like, and that might be leaning towards more Prevotella here in Kazakhstan, but then lifestyle also plays a big role. I mean, I haven't shown this result, but we also try to disentangle how much of influence comes from that, and Prevotella is quite an eminent member of the microbiome, so I think this could be something that we could answer together, I think, with the data that's coming out.
Yes, thank you. It's also very interesting for me to see what is in Kazakhstan.
Yeah. Thank you for the question.
Any question else?
Hello. I have a question for the Professor Uwe Simon. Simon or Simon? Sorry. Maybe it's actually a naive question. I have two questions. Have you looked at the role of ER calcium in the formation of those DNA traps? And the second question is, have you also looked or any evidence of this DNA formation depending on pH, for example, pH acidification? Because for sure you know that yes. The ER is the main source of the calcium.
Yes, you need also the intracellular calcium in order to get extracellular calcium, so this is a short answer. And for the granule proteins, you need acidification of the granules after activation in order to get degranulation. And you need some degranulation in order to get the granule proteins out of the cell.
But in that summary, if I remember correctly...
No, I didn't show any pH data. Didn't show this.
Okay.
But it's just an answer.
to your question.
Okay, I got the answer.
Else?
Hi, I have a question for Mohan. Thank you so much for the interesting talk. I was very curious when you mentioned that based on the data, you saw that accessory functions from virus could be integrated in their host and then maybe provide a better fit. I'm curious to know if there is publications on in vitro validation or if you guys have done something like that, in vitro validation of this observation in terms of fitness of the host and evolutionary advantage.
Yeah, it's a very, very interesting question, and actually we didn't conduct rat experiments for these deep sea viruses because most of their hosts are very hard or even cannot be cultured. But we conducted some similar experiments for viruses collected from high plateau hot springs, and the results validated that they do have this kind of contributions to the host metabolism. Yeah.
Would that comes from integration of the chromosome, or is the presence? Can you?
Most of them are not integrated into the chromosome. There are still circular elements aside from the chromosome.
Thank you.
My question also to Professor Kushugurova. My question is in your study you take samples from the patients, how many patients there were, and the next question is you find that Clostridium and Revatella populations are not so good in human microbiome. Just the question is because I'm also studying the atherosclerosis, and what they will be next your research in these bacteria populations. Thank you.
Thank you very much. First of all, our research is a part of the big targeted program, and it seems to me about 200 patients and a little bit more healthy control, but because we work with the heart center and the patients from entire Kazakhstan, so the healthy control too were collected from other Kazakhstan regions. Additionally, in this program, we are going to continue, and we're interested in some biomarkers like the protease of cysteines family, of course LPS, so we are going to continue just a few first preliminary data for this research. Yeah, and we are really interested because of we're interested to study the Pivatella species and even some maybe strains because of, according to our research, we have several autoimmune diseases, rheumatoid arthritis, spondyloarthritis. We work with the patients with Alzheimer's disease, and we observe that in all these patients we have a higher amounts of Pivatella. But it seems to me it's different because later we can observe the different type of immune reaction. Some of them for TH17 pathways, other, for example, cardiovascular TH18 pathways. So we would like to understand the differences. So we are going to continue. Thank you very much.
May I have a question to Lucas? Yeah, because actually my PhD project is also relevant to the industrialization of populations and their gut microbiota shifting. So, according to my previous analysis, I found that there is a kind of centralization during the industrialization between, or say across different populations. Do you see similar results in your study? Because I think you also studied many different populations.
Yeah, so you mean centralization in terms of convergence?
Yeah, something like that.
Yeah, so what we saw is that there is a much more connectivity of microbes in the microbiome, but the populations that we observed, quite the contrary actually, because there was a decrease in richness and of the industrialized population, so they tend to be more standardized in a way that they would tend to be similar to each other than when we map this to the non-industrialized, because there was, again, there's a bit of confounding factor there, but when you say non-industrialized, you have a much broader lifestyle background and genetics background as well than what was included in our study. So, but that would be interesting. I would like to, we can talk a little bit more about it and double check with your research and why we are finding apparently contradictory or different results.
Yeah, sure. Thank you.
Thank you.
Okay. Can I ask the question? My question for Professor Simon. Thank you for your presentation. My question might be out of your scope, but it's interesting for me. You know, like proportion of neutrophils and lymphocytes lead to the debate prognosis of the many of the cancer diseases, and I had the idea that it might be that this extracellular matrix might be scaffold for the cancer cells. It's like the in microenvironment of the many of cancers is they are releasing the calcium very high amount. It's like it might be its influence to metastasis too.
Yeah, there's published literature on this topic. We have not worked on it, but you have, there is more evidence. I mean, there's dispute, but I think there's more evidence that NETs could contribute to metastasis. So it does not contribute to cancer development or something like that. It's a clear defense mechanism, but when there's a cancer, there's a possibility that neutrophil extracellular traps could contribute to metastasis.
Might be it's like they are scaffold for the solid cancers, like to growing in the same places.
I just don't know the answer. Yeah.
Okay.
It's the same with neutrophil. If you find a cancer which is infiltrated by neutrophils, you don't know whether this is beneficial to the patient or whether this helps to grow the cancer. So there are different opinions. It may also differ from one cancer to the next. Same with the eosinophils, actually.
Okay, thank you.
Okay, just let me know if we have time, because according to this program schedule, we should have group photo, or we should go to dinner. Welcome reception. Okay. So we can do group photo. Group photo. Okay.
Зачем фото? Потому что там Кейтлин не успевает Шопан. Ну я же пишу в чат, вы что не читаете?
Dear colleagues, if anyone wants a group photo, let's go in front. Unfortunately, we won't be able to stay on top of the podium, but we can stay over here. I think not everyone was in the morning for the group photo, so it's a nice chance to actually get this photo done. Да. One more time for everyone who made a photo. Сейчас придет. А где фото? Фотограф где? No, maybe ladies will take place, yes, Инна Петровна. Шелпан, ну, как бы деваться некуда. Девочки, вперед. Вперед, быстро садимся все. Садимся, ну вы что, неудобно, нормально. Ну, Thank you, everyone. So we can now proceed to welcome reception in the atrium, together with sponsor exhibition and poster session.
Okay.
We will do this, no worries.
I will have to leave because I'm now at the...
And you are,
and you are.
Yeah, no worries.
So I'll be...